Okay, now we're starting to stream on YouTube. Okay, now then, accetta. We have 37 people online. We have, okay. Before I leave the floor to Professor Malatesta, I would just like to open the second session of this workshop, thanking again attendance in presence and online for their participation, thanking the speakers for coming here, and thanking all the people who helped, who contributed to this, the organization of this workshop. A special thanks to Professor Mancha and Professor Archer, Margherita and Filippo, who helped a lot in selecting the fantastic speakers we're having these days. Now let's share the screen. It's this one, Francesco. Yes. And Professor Malatesta will give a short introduction.
Can we remove this bar there?
Of course. Allora, that's it.
Good morning to everybody. Thank you, Beatrice, for the interesting, I love membrane proteins, and obviously also for the beautiful dinner last night. I will just briefly give some thoughts of mine concerning the change, and let's set this. Okay. Of course, membrane proteins are among the most fascinating structures in biology, and as we all know, by definition, this was Richard Henderson's definition, they are located at the interface between two aqueous compartments, such as, for example, the mitochondrial matrix and the intermembrane space in mitochondria in eukaryotes, of course, and or the cytosolic space and the periplasmic or extracellular space in prokaryotes, as we will hear also today. Of course, I need to tell you about very briefly what you know already. Since 1985, it was an important year because the first membrane protein, in particular the photosynthetic reaction centers from Rhodos Pseudomonas viridis, was determined by the Hart-Nickel group, in which we found this splendid structure with was a helical structure with all the cofactors, which clearly delineated the electron pathway in this fascinating protein. In a beautiful review, which is a little old, but I still valid, Richard Henderson classified these important proteins according to their function, and as you can see, this list will probably increase in the future. We have ion channels in pores, active transfer. Think about ATP synthase, mitochondrial and bacterial ATP synthase, protein translocators, and I was most interested in electron transport chain, and so on and so forth. And of course, you might know this beautiful website by the Stephen White, and as of today, actually this morning, we have in the protein data bank just a little bit of it in excess of 1,400 different structures. Okay, but of course we always must keep in mind history, and 10 years earlier, you may recall that Richard Henderson and Unwin published a seminal paper on the three-dimensional structure of the Halobacterium halobium purple membrane bacteriorhodopsin, which is a light-driven proton pump. Fascinating study. And he actually, probably he was visionary, because in a certain sense, it's sort of a you know, an anticipation of cryo-EM in many respects. In the central image, you see the parts of the three-dimensional structure, three-dimensional potential map. Let's see, yes, we're looking at here, showing a region of around 50 angstrom. I have to read the number exactly. And from thick spanning the membrane. And in this case, you will see a trimer which was typically found in these purple membranes, and eventually published a low resolution structure, which is this famous image published in this paper, was black and white originally, and which clearly showed for the first time that membrane proteins adopted canonical alpha helical structures. Although, of course, much later it was found that also beta battle containing proteins could be, nature could use them to carry out very many functions. Sorry for that. And I just want to close off showing a problem I still have after 37 years. This was a paper we were working at the time on cytochrome C oxidase, complex 4, the Atmungstegment mitochondria, beef heart mitochondria. And we were using a system which we upgraded very much, which was the incorporation of this protein inside artificial unilamellar phospholipid vesicles. And we characterized a bunch of papers, some 20 papers on the characterization of the system in terms of distribution of sizes of the liposome, sidedness of the protein, buffering power of the phospholipid bilayer, both outside and inside. And we managed to set up a protocol which allowed us for the first time to confirm what was demonstrated in mitochondria by Martin Wickstrom at Helsinki University, that indeed cytochrome oxidase is a redox-linked proton pump. So when we studied this system in the well-characterized system in the on the stop flow, we found that look at the open squares. We found that the kinetics of cytochrome C oxidation was biphasic, and actually, if you got rid of both the proton electrochemical gradient, for example, by both the membrane potential alone or the application gradient alone or both, the rate would increase. It was always exponential, but it would increase by a factor from 6 to 10-fold in these very tightly coupled vesicles. And this was in the same year of the Hartmut Pick paper. And of course, time went by, and about 10, 11 years later, the fabulous structure of the Yoshikawa group was published. Actually, it's a nice story because it was published in Science in May, in Science, I think, just to see how competition was tight. A week later, Hartmut Michel published in Nature the structure of the bacterial enzyme, which was very useful because in terms of simplicity. But the point is the following I want to make, and I want to show you now a very new approach, which I think is already a seminal approach. The problem being here that, of course, these effects we've been seeing by kinetics were interpreted in terms of a model in which there were a two-state model in which, there were two conformations of the protein which were populated depending on the presence of the membrane potential. Okay, so we were praying to find differences in the structure of cytochrome C oxidase. This is the beef heart, and so not surprisingly, nature is many times quite elusive. No matter what you did to the protein in the reduced state, in the oxidized state, in the presence of ligands, carbon monoxide, nitric oxide, cyanide, absolutely nothing significant changed. That's it. And there we got stuck, and we still are stuck to this problem. And that's why I was really pleased when I read this paper, which hopefully some of you know. It was published only 2 years ago at the beginning of the pandemic, in which for the first time there was a cryo-EM analysis of membrane protein in ELISA. That was only exactly as similar to the ones. Oops, what did I do? I inadvertently. Okay, and this was taken. Get rid of this. This was taken from an RCB transporter from a multi-drug resistant proton gradient-driven transporter, which was chosen by this Chinese scientist Mian Yang, which works in Princeton currently, and they were able to solve the structure in vivo, sort of in vivo situation. And of course, this was really, a breakthrough in the understanding. I don't know why I do this, but it does it on its own. Okay, and obviously it's in the author's ideas, original ideas, that this outstanding IAD approach can be widely used in the next future to, to cryo-EM analysis of membrane proteins. And I quote here what they write in their discussion. This lays out the foundation for cryo-EM analysis of integral and peripheral membrane proteins whose functions are affected by transmembrane electrochemical gradients and or membrane curvature. So have a look at this beautiful paper, and I hope this will, change things, because I think you all agree that studying a membrane protein, I mean, I'm sorry, but I hate these, detergents and these strange nanodisc. I don't understand, but eventually I think then we will have to end up by studying the structure in, in reconstituted systems. Thank you. And okay. And now we start the second session. Linda will share it.
Good morning, everyone. Let's wait a little bit. I would like to thank Professor Malatesta for this wonderful historical overview of the structural biology and biophysics of membrane proteins, and that introduced the sessions of this morning that are basically dedicated to to structures of membrane protein coming from eukaryotic and prokaryotic world. Since we are running a little bit late, I would like to invite the speakers of this session to be on time with their talk. And now it is a great pleasure. Are you ready? It is a great pleasure for me to introduce the first speaker of this session. That is Professor Filippo Mancia from the Columbia University Medical Center of New York, USA. Professor Mancia started his career as a structural biologist here in Italy, in Pavia, where he graduated in chemistry, and then he moved to UK at the MSc-LMB in Cambridge for his PhD. His postdoctoral work was carried out in the labs of Professors Wayne Hendrickson and Richard Axel at Columbia University, when he then became a professor as well as the Department of Physiology and Cellular Biophysics. Professor Mancia's research focused on the structure of membrane proteins and its relationship with their function, and for years he used X-ray crystallography as lead technique, but more recently he has been taking the advantage also of the revolution of single particle cryo-electron microscopy, and today is going to show us the structural basis of lipopolysaccharide biosynthesis. Professor Mancia, thank you. The floor is yours.
The floor is a big concept.
The chair is yours.
Thanks. So I will be masked. And first of all, I'd like to thank Bea for this kind invitation. I'm a frequent guest of Rome, and it's always a pleasure. I really feel Rome to be my second scientific home after the Milano Pavia access. And in a way, sometimes it's been together with the Milano-Pavia axis. So thank you again for hosting me. And thank you, Linda, for the superbly kind introduction. I'm really happy to be here. It's my second in-person meeting since the pandemic, and thank you all for joining. I'd also like to thank Professor Malatesta for essentially having given what I was going to do as a first few slides introduction. So at the expense of being repetitive, I will nevertheless go ahead. And I'm a structural biologist. I'm a membrane protein structural biologist. And it was in one afternoon of 1982 in June, in early June, when us Italians, I was a little teenager and getting ready for the triumph of the World Cup in Spain. But up in the hills of Erice, Hartmut Mikl stands up and shows this. This is the first diffraction pattern of the photoreaction center, and it is, as of records, the first diffraction pattern shown of a membrane protein. People stood up and gave him a standing ovation. This, from the diffraction, leads to phases, leads to structure. And as Professor Malatesta showed us, out came the structure published in Nature in 1985 of the photoreaction centers, making this the annus domini, or the year zero of membrane protein structure biology. Mikl, Diesenhofer, and Robert Huber were awarded the Nobel Prize a couple of years later for this work. And since this structure, the progression in membrane protein structure determination had years of lag time. And then came the first channels with Rob McKinnon, the first transporters with Eric Guo, and many more. And we have entered a phase of exponential growth in the field. However, despite this phase of exponential growth, and despite the importance of membrane proteins, there are 30% of the entire proteome are membrane code for membrane proteins, and they are the targets of over 50% of all approved drugs. They're only about 3% of the deposits. Now, the reasons for this come, and we have no time for this today, really come at every stage of the production of homogeneous material pipeline to produce the milligram quantities necessary for structural studies by X-ray crystallography. Until about a decade ago, the only and the most successful, if not the only, technique to obtain high resolution structural information. And the biggest conundrum, the biggest drama of membrane protein research really stands in the use of detergent, where, as shown in this central diagram, the environment of a membrane protein from a typical bilayer, which is shown above, to one in which of the micelle is completely different. This causes destabilization, as we all know, as well as the prevention, given this fear of heterogeneity around the membrane protein, creates a problem, often insurmountable at the crystallization phase. So we and others, and I link back to Brian's talk yesterday, spent in a way more than a decade developing methods to identify those membrane proteins that would have an increased probability of yielding structural information. So when you see here on the right that at every stage you lose between 90 and 70% of all your targets, this implies that for 1,000, this statistics here implies that if you take 1,000 random membrane proteins, you would only obtain crystals of 2 or 3 of them. And these were the initial crystals, which didn't necessarily lead to diffracting crystals. We spent time in developing methods to essentially improve the yield by about a factor of to identify those membrane proteins in a rapid and high throughput manner that would pass all the tests before crystallization. So we and others spent time in dedicating ourselves to the biochemistry of membrane protein research. And it was therefore another second eureka moment after the work of Hartmut Mikl, when about 9 years ago, to be precise, David Julius and Ifan Chen at UCSF showed that they could use single particle electron microscopy to solve atomic resolution structures of membrane proteins with their work on the trip channels. The defining moment of this was the fact that we had spent decades, 20 years, learning how to make the samples, and now the most difficult step, which is that of crystallization, was put aside. And not surprisingly, as shown in this graph here, as of today, as of a couple of years ago, the number of EM structures of membrane proteins has superseded that of X-ray structures. Not only, the improvement in the technique has allowed a series of evolutions that we talk about. You go into the cells, you tomography, you isolate low abundancy macromolecular complexes. So there's really, and from my perspective, there are 3 points. One is the fact that for single particle cryo-EM, you need microgram as opposed to milligram quantity. And two, that it allows you to deal with progressively smaller proteins and to analyze them in the lipid environment of, let it be a life we will see in the future, or more simply, of a lipid-filled nanodisk. So this is where we come in as a lab, where we have 2 main interests. One is to use structural biology to understand molecular mechanisms of drug resistance. And I think I have talked here in Rome of our work of the polymyxin resistance mediator ARNT. This enzyme takes a sugar, neutralizes a negative charge on a phosphate, and all the positively charged antibiotics don't work anymore. Or our work on the malarial transporter PFCRT, which mediates the resistance to chloroquine, the historically most used antimalarial. And the second interest is that of is more a biophysical one, of understanding how the membrane protein, and substrates which are lipidic are either enzymatically modified or transferred. And if you think about it, a lipidic substrate sits ideally in the membrane. And the reaction, an enzymatic reaction, for example, requires an aqueous environment. So how do you mediate? Where does the energy come from, and what are the elements of specificity? I have presented here my work. Oh, sorry, let's go back. I've presented here my work on the vitamin A transporter STRA6, and in a talk in the fall, I presented to you the work on the complex of a lipidated Wnt in complex with Wnt. I haven't showed you my more recent work on the omega-3 fatty acid transport MFS-E2A, but what I thought I would do today is show you our more recent work, which really brings together both that of the molecular mechanisms of drug resistance and the transport and the transfer or the modification of the substrate. So let's take a very deep, a very quick step back. We've heard a lot about viruses in the last 2 years, unfortunately, but linked to this, in a parallel world, is the issue of antibiotic resistance, which in 2019 caused over 1 million deaths and has been responsible in a more indirect way for about 5 million deaths worldwide. This is a big deal. And the crisis in the emergence of multi-drug resistant bacteria, which caused directly or indirectly, is due to 2 factors. One, the lack of investment in the field. In the last 30 years, there has not been a single new class of antibiotic brought to the clinic, and the progressive increase of use and of drug resistant pain against every one of the major classes of antibiotics. A major mediator, and we're now switching to gram-negative bacteria, which are the culprits of the majority of antibiotic resistance infection, is the outer cell wall. The outer cell wall is an impermeable barrier in a way. We'll hear more about it, how mycobacteria make it even worse, from Margherita in the next talk. And it's composed of a peptidoglycan layer shown here in the middle, and an outer layer, which is a mixture of phospholipid and lipopolysaccharide. The lipopolysaccharide is constituted of a lipid A anchor, shown in the right in black, a core oligosaccharides, and repeating units, a polysaccharide, very variable repeating unit called the O antigen. The LPS, and in particular the O antigen, are the major mediators of the host pathogen interactions. That is the element that is responsible for immune evasion, from both immune evasion and for the pathogenicity of the bacteria. It's, in crude terms, the first element that the immune system of the host sees when the bacteria is present. So not surprising, the biosynthesis machinery of the mature LPS is a pretty complex thing. It's essential for the bacterial, gram-negative bacterial. So on the left, so on the very left is the structure of the LPS. As I said, the core oligosaccharide and linked to lipidae, the O antigen. So the O antigen component, which are these repeats of sugars, is formed, synthesized by glycosyl transferases within the cell, and it is then flipped to the outer leaflet of the inner membrane in a series of pathways. It depends on the bacteria. It can be the what's called the WXY dependent pathway, a less characterized synthesis pathway, or with ATP, with an ABC transfer. On the other side, on the right, is the MSBA derived synthesis of the oligosaccharide. The two then have to come together, and the enzyme that does this is called WAL, or the O antigen ligase. The O antigen ligase transfers the O antigen here down on the bottom right from an undecaprenyl pyrophosphate. That's 11 prenyl units with 2 phosphates. It's a typical lipidic sugar carrier. So the O antigen attached to the undecaprenyl pyrophosphate is transferred to the outer core of the core oligosaccharide lipidae. By this enzyme of 44 kilodalton metal-independent inverting glycosyl transferase. Don't worry about it, we'll get to it. You knock out WL, no LPS can be made. Once the mature LPS is made, the LPT pathway transfers it from the inner membrane out to the outer membrane, where it can do. The involvement of two lipidic substances, the lipidae and the undecaprenyl pyrophosphate carrier, the fact of its link to mechanisms of drug evasion and antibiotic resistance had us focused on using structural biology to understand how WL works. So we used, as Brian showed us today, a structural economics approach, and we screened about 200 WAL orthologs to identify those that were amenable to structural studies. This shows some gels with and without TEP cleavage of a histidine tag of a subset of 19. It's a subset of the subset of 19 where our best candidates based on expression levels and stability in detergents. And we identified one from the organized Cupraviridis metallidurans, which, as shown by the size exclusion chromatography traces here, was the most stable in detergents used for structural studies. Now, the O antigen component is highly variable. It's highly variable within different species, within the same species, not surprisingly amongst different species. So this enzyme, when placed in a WAL knockout in E. coli, didn't work. So we had to go to the Cupraviridis and make a genetic modification of Cupraviridis to show that indeed the enzyme that bioinformatics had showed us to be hypothesized was WAL was actually doing its job. The gel showed here. We went brute force into crystallization, which failed. We placed the element in nanodisc, and the filtration shows different nanodiscs with different lipids, and you see a shift to the left because you have added mass in respect to a detergent Basel. But not surprisingly, given the 44 kilodalton integral membrane protein in a nanodisc, the 2D class averages from initial cryo analysis looked like discs, and we could see no membrane features. So we adapted a trick that we have used in the past, both for our work on PFCRT, the malarial resistance transporter, and our more recent work on the omega-3 transporter, MFS2A. And in collaboration with the lab of Tony Kosiakov at the University of Chicago, we provided biotinylated nanodisc with WAL, and we screened a synthetic library of rigidified FABs. This allowed us to identify a series, I think it was about 6 high affinity binders, which we characterized further here, shown on the left. We took the highest affinity of these these binders, and we reconstituted the complex. So we made the ternary complex of WAL, lipid film nanodisc, as shown in the gel filtration spaces in the middle, and in the gel, in the last lane of this gel. Not surprisingly, the fact that we added 50 kilodalton extra molecular weight, and most of all, a feature, a rigid feature, which we could use to align one particle from the other, allowed us to shift from those disk-only 2D class averages to these, where we could see both the clear domains of the FAB, and within the nanodisk, we could start seeing at the 2D class averages the features of transmembrane species. Flash forward a few months of data processing, we were able to obtain a structure, a 3.2 angstrom resolution of the FAB WAL complex, WAL nanodisk complex. And now it's working. That's great. So the video's late. And here shows the density map in which the FAB is in gray and the WAL trace is colored from blue to red, according to from N to C terminus. What does the structure look like? As expected, the structure is has a 12 transmembrane with quite a salient and 90 amino acid also region in the periplasmic place. What to note here is the presence essentially in the middle of the membrane of transmembrane helix 9, which is linked to this region of the periplasmic region I was discussing. So, and previous work on this enzyme, extensive previous work, had shown that this periplasmic region is essential. It's essential. So if we look at the structure a little bit more closely in terms of conservation, and here is a conservation plot on the left, and on the right, it's plotted on the structure with purple being the most conserved on the top, and bottom is the electrostatic. You can see that these enzymes aren't really that conserved. It weren't for a cluster of absolutely conserved arginines and a histone. So the arginines, not surprisingly, cluster in this region shown here in blue and in conservation here, which is, not surprisingly, at the interface between the soluble and the membrane. If we went and looked and we mapped the cavities of this structure, then the one region where the arginines are constitute one of the two cavity regions of the molecule. Now look to the right and let's just remember what the reaction that this enzyme catalyzes. It takes a large lipid A component and an undecaprenol with the antigen attached. We looked inside the cavity and we were lucky enough to find density within the green cavity here and in proximal of these four arginines, absolutely conserved arginines, we could interpret as being a partially ordered undecaprenol phosphate. We could see an ambiguous density for the two phosphates and for two phenols. We went back into the cupraviridis and we showed the deletion of three or four out of these arginines was absolutely deleterious for function. The same we recapitulated in the E. coli model. And the results of these experiments are shown here. So in our KIM analysis, we had picked up an obvious product, the undecaprenol. The tail is, well, the phosphate, the pyrophosphate is coordinated by three of the four arginines. The tail sits the first two prenols sit in a groove, which is shown in blue here, which is comprises, not surprisingly, hydrophobic residues. We used TLC to confirm that what we thought was undecaprenol pyrophosphate was indeed undecaprenol pyrophosphate. In this experiment, which is still ugly, caused a lot of havoc. And Francesca was probably laughing. There was some drama on this one. There's always drama. And then we used molecular dynamic simulations, shown here on the right, where we used what we saw in the experimental density, in the experimental map, the density map, to trace, to model the entire undecaprenol And we showed in terms of RMSD movement that indeed the first 2 prenuls in red on the graph and shown up here are kept rigidly within the structure, while the remaining 9 can wobble around in the membrane. So again, we've seen this many times. The enzyme, the elements that determine the specificity are the pyrophosphates. That's what keeps the ligand there, while the membrane is used to provide a stabilizing environment for the long lipidic tail of the carrier. We went back into our cryo-EM data and we noticed that we had a minority, still present class, in which there was no density for the undecaprenal pyrophosphate, shown here in purple. An overlay, so we had in one data collection, we identified 2 classes, with the undecaprenal pyrophosphate product and without. If we look at the differences, it is obvious. I hate this, these PCs. If we look at the distance, if we look at the difference between the ligand bound and the apo structure, the biggest distance, the difference is in the transmembrane 9, this transmembrane that goes outside. It is obviously way less ordered. And overall, the structure of the apo enzyme is of lower resolution, as if, not surprisingly, the presence of the pyrophosphate ligand gives stability to the structure. Some of the conformation of the arginines in the pyrophosphate binding site change, not surprisingly. And if we perform molecular dynamic simulation on the entire molecule with and without the substrate, we observe two things, that the regions that fluctuate the most are always the same. The periplasmic region shown here as region 3 and in purple, and the transmembrane helix 9. But moreover, the apo structure has an energy minima in a conformation shown here in blue on the right, which is more open in respect to the ligand bound, suggesting a conformational change of transmembrane 9 linked to the coordination of the pyrophosphate by the 3 arginines, which could be essential in shuttling the lipidic ligand in and out of the active site. This, however, leaves open the second and most daunting question, is given that one ligand we've seen, and it is the most simple of ligands, it has 2 phosphates, it's coordinated by the arginines, and it has a single polyprenol tail. The lipid A component is way more complex. It has 6 acyl chains, and it has the inner and the outer core and the outer core. So if we look at the cavities, we can see a second cavity in blue at the back of the molecule. And if we look at the back of the molecule by electrostatics on the left, hydrophobicity on the center, and by conservation, we can see, yes, that there is some hint of something, but we can't really tell that much. However, if we go back at the density, and we do focus classification on that region, we see that on the back of the molecule, there is a membrane deformation shown here. The membrane of the nanodisc deforms. This is due, we interpret, to 2 helices of WO, which are shorter than usual and have some charges. We take the model and we perform molecular dynamics simulations shown here on the right, and again we see a membrane deformation of about 7 to 9 angstrom in that. We hypothesized based on this that this might be the lipid A binding site. Molecular dynamic simulation in which we place lipid A there shows a site of contacts where these 2 shortened transmembrane helices, a potential site of contact where 2 shortened transmembrane helices are shown here in blue. Now, if we work it out, the distances could match. We're at the other side of the molecule, but the inner and outer core span about 25 angstrom and could be more than enough to span the 25 angstrom that distances the second cavity. However, obviously, with a massive ligand like lipid A, mutagenesis of single or double amino acids does lead to a decrease somewhat of activity, but it's not a cutthroat, clear-cut yes and no experiment. So the question mark remains. It does remain, but we also feel comfortable in proposing at this point a mechanism of how substrates enter and exit the active site of this enzyme, in which we see the transmembrane 9, an opening of the transmembrane 9, exemplified here on the left, and a cavity which comprises, as we'll see in the next slide, the active site for the undercaprenal pyrophosphate link or antigen. While the lipid A core oligosaccharide binds on the other side of the membrane, and the reaction and with the oligosaccharides spanning above the molecule into the active, leading to what happens in the active site. I will not go necessarily into the chemistry of the reaction, which is quite complex, but let me just say that crucial to activity and conserved in all WALs is a histidine. In this case, histidine 338 that we showed both in Cupra viridis and in an E. coli model was absolutely crucial for activity. So this structural biology and functional experiments allowed us to make a hypothesis on how the reaction might occur, in which the histidine abstracts a proton from the donor, from an OH of the donor substrate, and becomes the nucleophilic attacker, the cleavage of the O antigen bond after the pyrophosphate, in a way that the reaction is similar conceptually to that we see in traditional insoluble glycosyl transferases. It allows us to propose mechanisms of reaction. Last but not least is step back in evolution, and what is emerging as structural biology contributes structures of these GTC fold glycosyl transferase striking similarities. And here I'm just showing you our WAL structure here, and this is a transferase compared to the processive glycosyl transferases ROD-A. This is a crucial enzyme involved in making the peptidoglycan layer that sits under the lipopolysaccharide. They look rather the same. The reactions they catalyze are different, but they both use undercaprenal pyrophosphate as a ligand, and they also have absolutely conserved arginines. So we hypothesized that the fold with an external transmembrane, shown here for ROD-A in 7, might be the mechanism in which these polyprenol pyrophosphate ligands go in and out of the active site. Not surprisingly, maybe, our WAL, shown here in gray, has the 2 additional transmembrane helices, which are the ones that we hypothesize are the key mediators of the interaction with lipid A. And not surprisingly, if we're correct, the ROD-A doesn't have those 2, because indeed the substrate of ROD-A is a lipid. It's called lipid 2. It just has the undercaprenal pyrophosphate. It doesn't have the lipid A component. And if we go it in broader terms, we can see that the analogy also is extended to other GTC glycosyl transferases. And you can see, without going into too much details, a clear analogy with the these periplasmic amphipathic peaks shown in blue for the lipid-to-lipid glycosyl transferase RNT that we solved, and by the work on the other GTC fold glycosyl transferase PG. So at this point, I will jump to conclusions, and we've just to conclude, we've determined we use single particle KIM combined with antigen fragment binding technology to determine the structure of the enzyme that brings together the 2 components, O antigen and oligosaccharide lipid A, the core oligosaccharide lipid A. It brings them together, ligates them to make the mature LPS. We have in one data set 2 structures with and without undercaprenal pyrophosphate, the product. We can see a cluster of conserved and functionally essential arginines that we hypothesize are there to both coordinate the pyrophosphate in order for the reaction to occur and to get the polyprenol in and out of the active site. We make a hypothesis based on functional experiment, mostly on molecular dynamics simulation, of whether the massive lipid A might bind. And we've used an integrated approach, goes back to my presence in this meeting, to combining functional experiments, MDs, and structural biology to start to understand how this fascinating enzyme works. Fascinating enzymes that seems to have a trend, let's call it, in the world of GTC, glycosyl transfer. Integrated approach require massive collaborative approaches, and I love collaborative science. It's just keeps me going. So I'd like to extend a few thanks. The first and foremost is to not the heroine, the hero of the day is Khurram Ashraf, shown here with me. He is now a postdoc with my former trainee Vasilis Petru at Rutgers, and he's done this. This was his project. Done brilliantly. He's been assisted by close second Ri Nygaard, and I'm very grateful to Ri, who participated in the processing and analysis of the data. This work would not be possible without our cryo-EM guru, Oliver Clark, who is Francesca's PI. Our two labs work together. And I would also like to acknowledge Phil Stansfield at University of Warwick, who did all the molecular dynamics simulation, and Stephen Trent, my longtime collaborator, who did together with Anne-Catherine Ulleman, who made the genetic knockout in Cupraviridis, who did all the functional experiments. I would also like to thank COMPA, and Brian is here, for the initial structural genomics approach that allowed us to select this one WL that seemed to work, and obviously no no microscopes, no electrons, no structure. So I'd like to thank the Calabria IM facility. Thank you, and thank you all for listening.
Thank you very much, Filippo, for this talk that also show us how a small protein can be always studied by single particle cryo-EM, even if it's under the limit, which are considered the limit of the technique. And so if you have any questions, yeah.
Okay, Filippo, obviously fantastic job, but I was wondering, since you solved the structure with the nanobody, well, with the Fab, which might distort, is it binding to the loop, extracellular loops, which are so important for the position of the helix?
Yes, but it doesn't. So it binds to. There are 4 little helices up there, and 2 of them have been shown to be essential. It binds next to them. Obviously, we cannot exclude this, but we did perform molecular dynamics simulation. We use molecular dynamics simulation to answer your question. Data I'm not showing, and shows that the molecule, in terms of movement and in terms of how the substrate moves within, is essentially indistinguishable with and without the A good point, obviously. We pay a price. We always pay a price for the fact that we need this fiducial. I mean,
wonderful and complete work.
No, it's never complete.
So I have 2 questions. Is there some energy gradient for driving the reaction? I mean, is the enzyme active in the membrane or also in solution?
So there's 2 aspects. We have not tested activity in solution, because the activity is whether we can make full LPS or not. So I'm not going to go into a reaction like that, which involves adding components which are difficult to isolate in the detergent lysis. So we did all the activity assays in the membrane.
So
now wait. Now the fact that the environment, the membrane environment, is most probably essential for activity, but we don't have the data. I'm not saying that it's not active in solution, but I wouldn't call it necessarily a gradient. This is not a transporter nor a channel. It's an enzyme. So the energy will come from breaking and making chemical bonds in the right.
And the second question is there any human protein which have similar structure?
So the GTC fold is present in human. So the machinery to make N or O linked glycosylation, it can be more complicated. One enzyme or a complex of heterodimers, but the fold, there are similarities. It's in a way, the prokaryotic systems always tend to be simpler to analyze, but the fold is consistent, and the biggest theme is O and N-linked protein glycosylation. Okay. And again, in terms of simplicity versus complexity, the undecaprenyl phosphate linkers, the 11 prenols become the dolichol phosphate twice the size. Yeah. You have all the dolichol phosphate pathways in eukaryotes.
Thank you very much,
Camillo.
Thank you, Filippo, for
Thank you, Filippo, for the great work. And of course, this is very close to what I do. So I really appreciate all what you show. I have several questions, but so let's start. You captured this state with undecaprenyl pyrophosphate, but this is the product, right? Well, part of the product which leaves, right? Which should leave the enzyme so that a new undecaprenyl pyrophosphate comes. So do you have anything to say? How will it go away? Why actually are you capturing an undecaprenyl pyrophosphate bound state?
So we've seen this before. And it obviously the fact that we have 2 phosphates and 3 conserved arginines raises a very good point, which I do not have an answer for, is how does it?
Yeah,
I agree with you. It's an unanswered question, which is the reason why at the comment, we've mapped it all. No, we haven't mapped. This is a first glimpse of this thing. And what we need to do, we're doing, is actually to go into an organism like E. coli, where we can control more accurately both the product and the substrate, because we can get them, we can make them, we can manipulate them. And indeed, where the next step of this work is to transfer the knowledge and solve the structure of E. coli WAL, which is what we're doing. That's going to be able to answer us. But it's and even the molecular dynamics, they show that APO and let's call it product bound move in a different way. One's more flexible than the other, but it doesn't answer the question of how do you release the substrate, which I think is
And I mean, also related to that is we all know that in bacteria, these undecaprenyl pyrophosphates are just this is very common in also wall synthesis pathways, independently whether you synthesize O antigen or other biopolymers. And you show that the last polyprenyl units are maybe actually not even important, maybe just very flexible. But still, this is undecaprenyl is always there, So do you have, but maybe, for this enzyme, do you think is not important at all, or maybe the undecaprenyl, the full length will be important?
We've seen that before. Every time that we've seen a structure with a prenyl, the ordered region is always marginal. Well, the membrane uses the nature uses the membrane to stabilize the rest of the cell. So my interpretation is that in terms of the enzymatic reaction, when you have to take a sugar from the carrier and put it somewhere else, the specificity is derived by the sugar itself, by the phosphates. And the rest doesn't count. Well, it counts to be able to energetically transfer, for example, the sugar from the cytoplasm to the outer leaflets of the inner membrane. That's when nature has made it. It has made it 10 in mycobacteria, or 11, or however numbers in the dolichol.
And are you trying to capture the lipid A bound state? Because this is the obvious thing.
Yes, of course. So we are doing this, but again, it's to an E. coli system, which allows us to know which lipid A to use, because dealing with the cupraviridis puts there are complexities of both the lipidae and most of all at the antigen component. So we're shifting. The next step is to shift to E. coli and do all the states. I mean, try to do all the states. We do have, and I shouldn't even maybe say that, we do have elements of we do have a structure of ARNT, the lipid to lipid glycoside transferase in the presence of lipidae now.
Okay.
That's another enzyme. So it can be done.
Yeah. Okay.
It can be done again. Most of the times it can be done when you fortuitously
you bring
pick it up during
to,
the presentation as opposed to re-adding it afterwards.
All right.
Thanks. You're welcome. Microfono, maybe anche senza mascherina, maybe we can tolerate it. Yeah. So, okay. Let me just focus on one aspect. I'm not going to go into the other techniques, and obviously we're welcoming many techniques. One of my dreams is to use single particles to do time-resolved studies of enzymes. The problem is the bloody time. So you can mix the two components and then freeze a grid very quickly. But we're still more or less an order of magnitude out in terms of how quickly you can do it. So we're getting there. So we have a system called GTRB that takes a sugar link to it's actually a nucleotide linked sugar and attaches it to the kernel. And we're trying it on that. And that ultimate is actually the kind of experiments we'd like to do, but we're one order of magnitude time long as opposed to short. So yes, and then what you do is you collect, and you see what you have. And the mixtures, the recent developments in single particle can allow you to classify out the mixtures in a very elegant and effective way. But you need to be in the right time scale, and we're not quite. And the approach that, if you take a transporter, the approach that Professor Malatesta was presenting of Mian Yang, and there was work in Yale that done previously. It's fascinating also, but the technical challenges there are also pretty big because you've got to image bloody liposome. So there you might have the time scale because it's a transporter, but you have background and all sorts of problems, especially with small membrane causes. So that is, to me, when people say single particle is dead, I've heard that say we should go into the cell and just do tomography. I say, no, wait a second. We've been an explosion of structure. We haven't really understood how they work. And in order to understand for an enzyme, we need to see it. We need to see the reaction happen and trap it exactly as you say. And for a transporter, or for a channel, we need to image under an electrochemical gradient. That's like my little, 2 minutes of philosophy on the future. Thank you.
So much. And now we have the second speaker. Can you see this? So I would like to introduce our second speaker of this morning, which is Margarida Archer from the Institute of Chemical and Biological Technology, ITQB in Lisbon, Portugal, where she is the head of the membrane protein crystallography laboratory. Her research is focused on membrane proteins involved in cell wall synthesis, transport and signaling, as well as on proteins that participate in catalytic and metabolic pathways, which could be potential targets for drug design. And today, she is going to present the intricate mode of action of arabinofuranosyl transferases in mycobacteria.
So first of all, I would like to thank Beatrice for the great workshops she's put together. And it was lots of work. And I have the difficult task of talking after Filippo, because it's always fascinating to, to listen to him. So I will start from the end with the acknowledgement, so that I just stress the work involved and the people involved. And mainly this was pushed by Filippo. We had this brilliant PhD student, so Yang Zi from Colombia, and then José Rodrigues, that also were very involved in this work. Then, of course, I will show you some cryo-EM structures. So we have the Siemens Electron Microscopy Center. It was where the data was measured. And then at some point, we needed to grow the proteins at the natural environment. So we had to go for growth in Mycobacterium. And so we had the help of the Faculty of Pharmacy in Lisbon to manipulate and make the training on this. And then there was some microbiology studies done at the University of Alabama, and then synthetic compounds made at the University of Alberta. So it's really, and I would also like to stress here the role played by Prometeus, because we were allowed to send students, namely José Rodrigues, then later Federico, to these long exchange visits in the US, and this was then made available by the Prometeus. Then before I will present the structure of the Mycobacterium, I thought, especially also because I think there are students here, to say that science is also made a little bit of serendipity. And this was just to say that at a certain point, we were working with these CDP alcohol phosphatidyltransferases, and it was the first structure to be out. We were very happy. We were writing, preparing the paper. And at a certain point, the biochemist that was working in this project received an email from Filippo saying, oh, we have this structure, but we would like to learn more about its function. And then it was a shock, as you imagine, because you always want to be the first, because being the first or being the second makes all the difference in terms of which journal you published, and especially also for students, what for their CV, for their later applications, it makes a difference. And so I had met Filippo just in a garden conference during lunchtime, but it was just this trivial conversation anyway. So we were just thinking, okay, what shall we do? And Elena did not reply for some time because we were still trying to figure out how to go, what to do. And so then we just decided, okay, let's just open the game and tell him we also have a structure. And then out of the 100 structures that are out there, it was the most similar one to ours, you know, because there are several others. You have a broad range of structures, but it was almost identical. As you see here, then just the orientation is different. Then we just decided, okay, let's try and it was years of work. So let's just try to make it work for both. So we were more or less saying, okay, I'm going to try to submit to this journal. Okay, we are going to try to submit to this one. And then after a certain point and a few refusals, then we said, okay, why we don't write to the editor and say, we have here two very similar structures. One had more functional assays, the other had ligands that we do not have. So we tried to make it, in a sense, it was complementary. And so, okay, let's go for independent revisions. but then try to publish just back to back. And then it works. And then it started actually a nice collaboration because we were also having some other targets. And that's where the story of this Mycobacterium membrane protein structure happened. Just so that then also another time, it happened almost the same thing. And then we also decided to get in touch with the other group, and then it also worked. And actually the editor was quite surprised, saying much better just to try to arrange things, that this was a win-win situation because we both managed to publish well, and then it also worked. So if possible, instead of competition, just try to organize things. It's much better. So so maybe now I'll just mostly skip this one so that we try to gain some time. But I'm always surprised that in terms of, if we look at Mycobacterium, it is estimated that 1/3 of the population has latent TB, because only around 10% of this latent TB goes into activity and affects mainly the lungs. So there are these first-line drugs to treat TB. The problem, or one of the problems, is this is a very long treatment. It takes between 6 to 9 months, and most of the people, when they start feeling better, then they just don't complete all the treatment. And this is causing a lot of resistant, Mycobacterium-resistant strains to appear, not only to one drug, but to several, and now to what it's called the extensively multi-drug resistant bacteria. And so, of course, this is causing around almost 2 million deaths per year. So we then decided to study this class of proteins that is involved in the synthesis of Mycobacterium cell wall, which is a complex structure. It is made mainly of this peptidoglycan, arabinogalactan, and then the mycolic acids. And in reality, this cell wall makes a protective and low permeability layer, which allows them to survive in harsh environments, and it also constitutes a barrier against lots of threats. So because these enzymes are known to be essential for Mycobacterium, of course they would be good. They would be druggable. They would be good drug targets. So I'm going then to focus on this arabinofuransyl-transferases family. There are 7 identified families, the EMBs and then the AFTs. The EMBs are putative agents, targets for ethambutol, which is one of these first-line TB drugs. And then these AFTs constitute new TB targets. And then I apologize, but apparently we forgot to do the share screen. I'm sorry. So you haven't been. No, okay, sorry. No, nobody's fault. Okay. That's it. Okay, thank you. The apologies again. That's it. Okay, great. Okay, so then they act in different places. They can be for polymerization, then branched, or in linear addition of these sugar moieties. But now it's not moving. So as Brian already mentioned, there is this big pipeline between selecting the targets and then getting some protein. So here is just to show you that out of almost 100 targets that were chosen for this seven families of these glycosyltransferases from 14 different Mycobacterium strains. And so out of almost 100, then half we were able to clone half, then express another half, and then so we had about 8 proteins that were able to be purified, and then 6 to be solubilized. So out of these, then there are already 2 EM structures out, and others are underway. And this was done, part of this work was done at Compa. So then we went also back to the lab, and then we also divided a strategy using then the 96 well plates, and then to go from the growth in very small scales into the purification, and then try to analyze the results and select the best candidates which to go for larger scale purifications. And then so I'm going to show you a bit the results in terms of the AFTD targets. So we had a few conditions, trying different temperatures, medium, time of induction, and then by selecting the best conditions, then we moved into larger scale purification. And before then going into the structure, just to see what is AFTD doing. So it is transferring a sugar moiety, so an arabinosyl residue from this decaprenyl arabinose sugar donor to acceptor, which is on the branch of this intricate polysaccharide membrane. So it is not yet known where it is the addition taking place. There are several studies, but there's still some controversy. And then we also wanted to have an idea in terms of what is the sugar acceptor. Is it branched? It has longer, so it seems to have longer preference for some longer, or better preference for some longer glycans. And so a glycan error analysis was done. And of course, we always want to get the ligands in the structures and see the differences. So in terms of functional assays, we tried to make a knockout, but it did not work. So there was this conditional knockout that was done, and that it is inhibited, or the expression of the gene is inhibited by tetracycline. And so what we see there is when inhibit the AFTD gene, then the growth rate of Mycobacterium, it's very effective. And so if we then see in terms of the growth in plates, we see that when we have the tetracycline, and so the gene is inhibited, we also see some differences in terms of the clumping of the cells. So in terms of the purification, different detergents, amphipoles were tried, and then at the end, then the protein was incorporated into the nanodisc. And just to call your attention that out of 10 liters of culture, we only got at the final hue as the protein purified and incorporated in the nanodisc, it was around 20 micrograms. So you could only go for EM, and these yields in crystallization, it is virtually impossible. So then it was the normal cryo-EM pipeline that was done at New York. And then, so in terms of the structure, we see that we have this, so it is a long protein. It has 1,400 amino acids. So we have this transmembrane domain where we have 11 transmembrane helices, which are here displayed in blue, which are characteristic of these glycosyltransferases, the GTC superfamily. And then we have some 4 extra helices, so a 4-helix bundle, which is here represented in red. And then we'll go, we'll have a look later what it's doing. And then we have this soluble domain harboring these carbohydrate binding motifs. So And then we have here something attached that we did not know what it was. So in terms of the soluble domain, we have identified these 3 carbohydrate binding motifs, and actually they are quite superposed with some lectins and other and this is this jelly characteristic, this jelly raw fold. Then in terms of the active site, this is an enzyme, so the first thing you look at, you want to see the active site. We see that it must be in the interface of the membrane with the periplasm, and we have the decaprenyl, the sugar donor, which is very hydrophobic. So the reaction has to take place at the interface, and then by looking at various sequence alignments, we see that, of course, as expected, around this site, we have lots of highly and strictly conserved residues. So we did some mutations also around the putative catalytic site, and we see that, of course, all of them then have a big influence also on the growth of the Mycobacterium. So in terms of then when the model was being built, there was some extra density, and we did not know what it was. We did not add anything to the purification. So we then identified by mass spec that this extra density belonged to an acyl carrier protein. So we did the expression of TFPD in E. coli. So we then identified that the E. coli ACP, it was just hanging there. So in order to make it more physiological, and then to see whether also if growing the protein in Mycobacterium, whether that would make a difference or not. So here we use the Mycobacterium smegmatis because it's like the model of the Mycobacterium tuberculosis, but not it is not pathogenic, and so we can use it in we can grow it in lab conditions. And so what we found out is when cells are grown in Mycobacterium smegmatis, then what we get here is the ACP from Mycobacterium smegmatis. Now, if we do some sequence alignment of these ACPs, we see that the ACP from Mycobacterium tuberculosis and lepra have this extra C-terminal longer tail, and this might also be related with so with a type of compound that is bound to this to this phosphopentate. So ACPs are 4-helix bundle that have this prosthetic group, this phosphopentate that is covalently bound by a serine. And then at the end, it has this sulfhydryl group that may bind to a cargo protein. And then we see that when ACP interacts with its cognate protein, and there's a very conserved zone in helix 2, then it is able to flip its chain from its hydrophobic groove into the cognate protein by then releasing its cargo. And so, of course, they are very promiscuous because the ACPs can interact with different proteins, and their role is well known on the synthesis of fatty acids. So ACP is either autonomous or it is in the mega complex of the fatty acids synthetic proteins. So here the big question was, what is the relevancy of the ACP? Why is it associated there? Actually, most of the times this interaction is very transient. So it was quite a while until complexes of proteins with ACP were found, because it just binds, releases the cargoes, and then just gets out. So here we see that this phosphopentate, this prosthetic arm, has already flipped from the ACP into the AFTD. So it is not in the hydrophobic groove of the ACP, it is already flipped into the hydrophobic groove of the AFTD, mainly surrounded or stabilized by hydrophobic residues. And then, so we see here that this is the region of this extra 4-helix bundle when we compare the sequence of the AFTD with other glycosyl transferases. And so then we did several mutations around the hydrophobic groove at the AFTD where the prostatic group is bound, and also in the interacting zone between the AFTD and the ACP. And then we found that, and then we had also truncated or a truncated version where this 4-helix bundle was excluded. And then we see that either these or then one of the mutants that is on the interface of the AFTD with the ACP made a big difference in terms of the growth of Mycobacterium. And so we decided also to go and have and try to characterize their structures. So we see here that in a catalytic mutant, and then in this mutant on the interactions between ACP and AFCD, we also see these different growth when the gene is inhibited. Then we see that in the electron density, we were expecting not to have the ACP bound again, but it's still there. So it's still, although much more flexible compared to the wild type. And then mainly we see differences at these regions from the two classes of the mutants identified with the wild type. And so we then see that there is a loop that is before in the wild type disordered that then becomes ordered in this mutant. And then we are still doing lots of assays in this respect with the ACP, but we sort of proposed eventually a regulatory role for the ACP, because we see that if the ACP is not bound, then there's this loop that gets ordered, but impairs the access to the active site. So here would be then the protein either binding. We do not know the order of the binding, whether it's first the sugar donor or the acceptor. And then when ACP is bound, it would work as an inhibitor of the enzyme. Of course, here we have the structures of the mutant. Here it's the structure of the wild type. So now we already have double and triple mutants where we see that the ACP is no longer there. And so the idea now is to get the structures of the AFTD, but then expressed in Mycobacterium, so that we also have the protein in a more native environment. So in terms of publications, we have a nice publication at with the structure and this information on molecular cell. And then also further studies were done to get the structure of the EMBB, so which is a target with the ethambutol. Lots of trials were tried to get the ethambutol in, but again, I think having the protein expressed in E. coli or in Mycobacterium, it makes a difference. But anyway, it allowed mapping of many resistance that are already in the literature and that are known to confer resistance to ethambutol, and several of them or most of them are near the putative active site. And there's a very one that is very prevalent, which is a methionine that is really on the pocket, and you can see it very well that when you mutated it, there's different interactions are taking place, and then ethambutol gets resistance in these mutants. Then more or less by the same time, although we did not know anything about it, there's a Chinese group that were also working on the structures of the EMB, the TMBs, and they had really a great job and very nice structures of several complexes of the EMBs, hetero or homodimers, and then with different already with different substrates, and then here with the ethambutol. And so here are the structures that were solved, and that we see that the ACP is in fact in all of them, although they also did not have a role for the ACP yet. So I think this is still a very intriguing, very interesting question. Actually, what is unexpected mainly, it's the most interesting things. So ongoing studies is to elucidate the role of the ACP. We are also trying to establish an activity assay based on NMR to allow for activities, to screen the enzymatic activities, to try to look for inhibitors. The problem is that these substrates are not commercially available. They have to be, so they are being done chemically. And then also one of the questions is, okay, we do not know exactly what the substrate is. So we are building different compounds with different lengths in terms of arabinose, also with branching or linear, and also at different spices. So this is ongoing studies. Although also here the problem is the sensitivity. So we have marked the sugar acceptors, and then we are now working on the assay. So also there's others smaller AFTs that are ongoing. So using, as Philippe already mentioned, either the antibodies, or in our case of the AFTA, we had with Brussels, the student went there to produce antibodies. So you screen for the better ones, the better candidate, the best candidates. And so now we have a mega body to go with the FDA, which is around 50, 60 kilodaltons, but now with the mega body, you just have size enough, it's big enough to go for the EM, although we are also having some problems in having good samples to go for the EM, but it's also work in progress. Okay, and I think that's it. Thank you.
Thank you very much, Margherita, and sorry again for all the mess you have during your talk. It was actually very, very interesting. There are any questions from the audience? Yes.
Thanks for the great work. These enzymes really are enigmatic, especially because of the substrate, and I understand the difficulties in getting the substrate in bound states. So my question is regarding the ACP, which I think is one of the most interesting things that you see, right, in the structure. So, I mean, why will the cell will like to switch the enzyme on and off with ACP? So, what is the reason?
Yeah, first we thought, okay, maybe because ACP is shuffling acyl chains for the fatty acid synthesis. So we thought, well, and we have many examples where it's just bringing the cargo in. So on the density you cannot see. Also then also on the other structures that was solved with the ACP in expressed in Mycobacterium, apparently there's also no cargo in. And then if we look, so these 4 helix, these extra 4 helix bundle are quite far from the active site.
Yeah.
So, and because then we saw that on these mutants, although still with ACP, but much disordered, there was this ordering of the loop that would prevent the access to the catalytic site. We thought maybe it can have a regulatory role, but I think we really need to get the structure on the triple mutant, double mutant, that we do not have the ACP, and then see in Mycobacterium what's happening. So this is one of the main goals now. And so because of this ordering of this loop and the hinder to the active site, we thought it may have a regulatory role, although it seems too complicated just to inhibit the protein. And usually nature goes to the easiest path. But so this, I think this is one of the most interesting questions we have right now. Well, of course, we are also going to try to get the whole picture of the AFTs. Yeah, but
Great work. Thanks.
because the assays are also tricky. We are trying mass spec, but then this is a 10 kilodalton in a 150 kilodalton with the association with 150 kilodalton protein. And so this is making things a little bit more complicated. We hope to have a clearer answer in the future.
Thanks.
Other questions? Maybe it would be also interesting to have a substrate site to see how this change with information actually it prevents the binding.
Well, so of course we tried. Then the question was, is it nanodisc or is it something hindering the access of the substrates? For instance, on the MBB, also ethambutol was tried, of course. But what we see is in the other structures that were solved, ACP is still there, and then you also have some of them also have the substrates, namely the decaprenyl, the DPA, and something, and it's still there. So, but of course, they also do not have a role or an explanation for that yet.
So we thank again Margarita for her talk. And now we move to the third and last speaker of this session, Professor Camilo Perez. Ah, sorry. Does it write it on the chat? I cannot find it. Okay. What should I do? Okay. Margarita. Okay, thanks. Okay, sorry. So maybe we can move to the next speaker. Worship. What should I do? Should I do? Okay. Thank you very much for your assistance. So I would like to give you just a short introduction of Professor Camilo Perez, who is coming from the Biozentrum University of Basel, Switzerland. So Professor Perez graduated at the University of Colombia in Bogota, but then he took a PhD in structural biology and biochemistry of membrane proteins at the Max Planck Institute of Biophysics in Frankfurt, Germany, where he also spent some years after his postdoc. Then he moved to Basel, where he is now professor and head of a research group that studies the molecular mechanism of cell wall membrane proteins. And this is also the object of his seminar of today, in which he will present the mechanism of cell wall transporters involved in teichoic acid synthesis.
Thank you, Linda, for the kind introduction. I'm really enjoying the meeting. Thank you, Beatrice, for the invitation. And yes, dinner last night was really delicious, so thank you for that as well. And I don't have to introduce any longer the problem of antibiotic resistance or how important are membrane proteins that participate in cell wall synthesis as drug targets. I think this is clear for everybody. So, but I would like to discuss about a particular membrane protein which is involved in teichoic acid synthesis, and it's very important for a certain pathogen, adaptation of a certain pathogen. And let's start by thanking the people that actually do the work in the lab. And today I'm gonna discuss the results of the PhD thesis of Bing Chang and Elizabeth Lambert, which graduated last year. And so this particular protein is actually important in adaptation. So what is adaptation, and how do we visualize it in you know, like in the animal kingdom? We see in different animals different kind of coating adaptation, especially. For example, that gives a protection, warning colors, or camouflage. Right? So these are different strategies that nature has used, or animals have used, to adapt just to different environments and to protect themselves. And similar things are happening in the bacterial world, where adaptation by coating also happens. And we have, for example, pili, which help bacteria to attach to different surfaces. We have flagella, which help bacteria to swim in different environments, or biofilms, which protect communities of bacteria. And the one that we like is the most are these cell wall, right? Which coat actually individual cells and protect them as well. But if we look in more detail in the cell wall, and in this case, this is the cell wall of a gram-positive bacteria. It's very different to what you guys what we have been discussing now in the morning. We don't have an outer membrane, so that's the main difference. So if we look at this cell wall, there are multiple different kinds of biopolymers. And all of them help the bacteria in adaptation in different ways. For example, we have capsular polysaccharides, right? They form the capsule, and by changing the composition of these capsules, then bacteria can actually be classified in different serotypes, and they also contribute to the evasion of the immune system. We have also teichoic acids, which are these biopolymers that you see here, anchored to the peptidoglycan or anchored to this glycolipid in the plasma membrane. And by changing the composition of these biopolymers, bacteria can change actually the polarity of the cell wall. And this helps them to hinder, for example, penetration of antimicrobials or different compounds, charged compounds. And of course, we have peptidoglycan, which in gram-positive bacteria is a very thick layer, much thicker than in gram-negative. And this helps the cell to maintain turgor pressure and, of course, protect from cell lysis. So in the lab, we are really interested in understanding membrane proteins involved in synthesis of all these biopolymers in gram-positive bacteria. And so there is this list that I'm pretty sure you have seen before, which is listed or was made by the WHO, where we have different pathogens which throughout the years, they have been showing resistant to antibiotics. In the lab, we dedicate our time to investigate, especially Pseudomonas aeruginosa. This is a gram-negative bacteria. Today, I will not talk about it. We also study Staphylococcus aureus. This is the one we are going to discuss today. It's an opportunistic skin pathogen and is very, very spread in hospitals, colonizing different clinical instruments. Is also a commensal bacteria in the skin of 30% of the human population. And we also investigate Streptococcus pneumonia, which is the pathogen that causes the main cause of pneumonia and meningitis globally. As I mentioned before, we are going to discuss just about Staphylococcus aureus today, and specifically about teichoic acids synthesis, just the pathway of teichoic acids. As I mentioned before, we have these two different kinds, teichoic acids anchored to the peptidoglycan, and lipoteichoic acids anchored through a glycolipid to the plasma membrane. And there are multiple studies on how important are these biopolymers for bacteria. And we know, just in this brief summary, that they are very important for host colonization, to interaction with the host cells, also in immune evasion, as I mentioned before, also in resistant to antibiotics. And if we look at the membrane proteins involved in the synthesis of these biopolymers, there is always something that repeats itself. We have always transporters, which usually accumulate precursors of cell wall synthesis by importing them. We have lipid transporters, or the so-called flipases. And this is because many, or as you have seen, most of the substrates that synthesize the final biopolymers that are required for synthesis of the final biopolymer, they are assembled on lipid head groups. And we have polymerases, which, as we have seen before, they are super important to actually the synthesis of all these polymers. And my lab is specifically targeting these 3 proteins, but today we are just going to discuss about flipases. So discussing about flipases, we need to come now to tell a little bit about lipid transporters and what we know about them. So they have been classified broadly in these 2 classes. We have scramblases and flipases. Scramblases are these type of proteins which actually disrupt asymmetry in the membrane. That means that when working, they will act as lipid channels that will just equilibrate lipids because they will diffuse across the leaflets. So at the end, you will have a membrane where the concentrations of the lipids in the internal and outer leaflet are equilibrated. But we have this other kind, flipases, which actually generate asymmetry. That means that you need energy to do this work, to translocate lipids in a vectorial manner. So we have these 2 different kinds, P4 type ATPases and ABC transporters. Both of them uses the energy release from ATP hydrolysis to translocate lipids. However, according to what we know today, they do it in very different directions. P4 type ATPases will translocate lipids from the luminal or extracellular side to the cytosol, and ABC transporters the other way around. And then we have this other kind, which is the one that we are going to discuss today, which are flipases that also work or translocate lipids in a vectorial manner, but they are more like a secondary active transporter type. These are the ones that we investigate, but actually one of the most interesting things that we like to investigate in these proteins is what is the driving force? What is the ion that is coupled to this process, if there is any ion coupling at all? And this is because that particular ion is very important, according to what my research has been showing so far, is important for this bacterial adaptation to different environments. So because we want to discuss about the mechanism of these proteins, let me tell you first about the 2 different mechanistic models that have been proposed for this type of protein, so for lipid transporters. We have the very first one, which is so-called trap and flip model, aka classical alternating axis, although I don't like to call it classical alternating axis because actually there are differences to how a regular transporter of a water-soluble molecule works. So this one here, we have just trapping of a substrate and release on the other side of the membrane. Yes, but what it implies, if we look at this example, this is what has been published by Tom Waltz in 2017. Here we have an ABC transporter. This is MSBA involved in LPS synthesis, and this will trap the lipid A substrate inside the cavity. It will release it once this little gate open. It will release it into the membrane. So this is the so-called trap and flip. And we have, on the other hand, the second mechanistic model, which has been proposed, that is the credit card model. So this is very different because here only the head group will cross the hydrophilic pathway in the protein, while the lipid tails will remain embedded in the membrane. Now you may think, okay, but why is this important at all? Well, I think it's very interesting, but also it is very important if we want to target these proteins with drugs, because if the protein just recognizes the head group, it's a very important piece of information if you want to design a drug, right? But if it also recognizes the lipid tail, it's also very interesting. Maybe you want to actually couple your drug to the lipid tail, and because in this case it doesn't recognize it, then maybe that's a good strategy to actually make a drug as a membrane. So, and there have been many different proteins that have shown this credit card mechanistic model, like for example this one right here, another ABC transporter. This is some work that I did in the Lohar lab in 2015, and here we have this ABC transporter involved in N-glycosylation. And here we have the lipid actually being recognized on the surface of the protein here. And when this hydrophilic pathway opens, the head group will cross. And that's a credit card type of mechanism. So the transporter or the flippase we are gonna discuss today, as I mentioned before, is important for synthesis of this lipotecoic acid biopolymer, the one which is anchored to the plasma membrane through this glycolipid. And the transporter itself is right here. We can see the 3 different steps that I mentioned before, repetitive among these different synthesis pathways. We have a precursor assembly, which is the glycolipid. We have a flipping of this substrate, and then we have the polymerization. In this case, is an LTAS protein, which will recognize the glycolipid and a PG lipid and synthesize the whole polymer. And of course, the transporter is central in this biosynthesis pathway, therefore, is a very interesting drug target. It translocates the lipid anchor. And the very first thing that we started to do when Bing joined the lab for her PhD was to do actually something suicidal. If you think about what Brian was telling us yesterday, what they do at Compa, because we actually take this one protein from this one pathogen and work with it until we get the structure. So that's, and I have to tell you, I mean, we dedicate like about a year or 2 to actually go just from optimizing purification, size exclusion profiles and so on until we got to this stage to have this great profile and also very nice looking crystals. Well, that's the thing. That was 2017. So crystallography was still a thing. And although nowadays we just do cryo-EM, but well, crystallography is useful as well. Well, Bing grew these crystals, these pretty nice crystals that diffracted to 8.5 angstrom resolution. So that was hard, right? and this was hundreds of crystals that diffracted to 8.5 or 12. So nothing could, take us away from that problem except for doing annealing. So that means that you will warm up your crystal a little bit for a very short period of time, expose it once again to X-rays, and then see what happens. And what happened is exactly this. They went from 8.5 to about 3.3, 3.2 angstroms resolution. So this was the way to go. Very enigmatic. It was just desperation of the night shift that took there, but it got us the structure, which we solved actually not by molecular replacement, but by heavy atom derivatization. So the structure of the protein looks like this. This is an outward facing state, right? And at the time, there were no structures of flipases from this particular fold. It's the major facilitator superfamily fold. So this was the first structure of a flipase from this one. And the very first thing that we noticed is that the cavity, the central cavity of this transporter looks very different to any other MFS transporter crystallized so far, solved so far. The reason for that is that the N-terminal domain of the protein right here encloses this surface, or what we call it now, a hydrophilic pocket, where you have multiple charged and hydrophilic residues. And the C-terminal part of the protein encloses this cavity that is very hydrophobic. As you can see here, all residues in this cavity are hydrophobic residues. So this was very particular. We compare with all the other MFS transporters crystallized. None of them have this feature. So later, Filippo Mancia's work on MFSD2A revealed actually a very similar characteristics. And now you have to see that this actually is very important, because this is a human transporter sitting at the blood-brain barrier, which will help the cells to translocate these omega-3 fatty acids bound to LPC. And so this is a very different system, but it's still the proteins look very similar. Like you can see here, the C-terminal domain of the protein encloses a hydrophobic cavity, and the C-terminal domain exposes a surface which is more hydrophilic. This is the same you can see here when we compare side-to-side LTAA and MFSD2A, and the same goes right here. So we of course we were really struck by this and amazed by this. So we wanted to go deeper into understanding the mechanism of these proteins, because now, okay, it's much more significant than what we talked about at the beginning. But of course, to understand how these proteins work, we had to not only have outward facing states, but also other states like the inward facing states. We tried with crystallography at the time when Bing was getting crystals, and we failed miserably. So we went for computational approaches. And the very first thing we did was to try to get these inward facing states by a repeat swap. And I'm not going to go into detail, but basically you can model an inward facing state of any transporter because we have inverted repeats. So we know how they should move in principle. And later on, well, while we were doing all this, AlphaFold came out and AlphaFold was released. So we say, okay, let's feed the sequence of this protein AlphaFold and see what comes. And what came here actually was an inward facing state. So they look a little bit different. This is actually much more open than this one. And this is actually what we revealed in MD simulations. These were done in collaboration with Gerhard Humer at the Max Planck Institute of Biophysics. And these inward-facing models, also in comparison to the outward-facing models, looked very stable in a membrane for the time lapse of the MD simulation. But of course, I mean, we wanted to validate this with real experiments. So that's the PhD work of Elizabeth Lambert, which designed mutants based on these inward-facing states and as a control also of the outward-facing state, where we replace residues here with cysteines in order to have a cysteine cross-links, right? Or disulfide bridges. And this is the results that you can see here. This is cross-linking of these proteins or these mutants in membranes. And when you don't have the cross-linking agent, you can see this one here, which means that there is reaction with a maleimide coupled to a PEG molecule. Therefore, there is a shift in the gel. But when you have the cross-linking agent present, this disappears. This means that the cysteines have reacted and they have formed disulfide bridges. So all these mutants actually validated these particular inward-facing states. We did the same in detergents. In this case, we use mass spectrometry, where we see in the absence of the cross-linker, we see the peptides. In the presence of the cross-linkers, these peptides disappear because, well, they have been crossed. So we validated this, and then we wanted to answer multiple different questions. The very first is, how is the cavity looking like in these inward-facing states? Now, remember that in the outward-facing state, we have the amphipathic cavity, and when we do docking of a lipid, the lipid will put this, will show the lipid tails in the hydrophobic pocket and the head group in the hydrophilic pocket. If we do the same with the inward-facing state, the model, we see a very similar thing. We see the cavity, which is hydrophobic, the C-terminal hydrophilic here down below, and the lipids will dock in a very similar manner. Lipid tails up, head group down. So this was very revealing that on how this protein binds the lipid. But of course, again, we wanted to confirm with real experiments. So what we did was a mutagenesis analysis of both pockets. We start by the hydrophobic pockets. We can see here all the residues involved, and some of them are highly conserved, and some are actually not conserved at all. And the first thing that we did was to try to make this pocket less hydrophobic. So we introduced a bunch of residues which are hydrophilic, like this right here, and we see an impact, a clear impact on flipping activity of this protein. And if we have these different mutations now in the pathogen itself, we can see also for multiple of them that the pathogen doesn't grow any longer that well. So we believe that the hydrophobicity of this cavity is indeed very important for, we hope, as our data indicates, binding of the lipid tails. We did the same for the hydrophilic cavity. You can see here the residues. These, on the other hand, are actually highly conserved. And we make the cavity less hydrophilic, and we see actually a great impact on flipping activity for most of them. And in the pathogen, we see that it doesn't like it at all when these particular residues are replaced, exchanged for alanines. Now, we did a very cool experiment, which is actually to have in these lipid or flipping reactions to have an excess of gentiobias. And this disaccharide has the same configuration or the same conformation is of the head group of the lipid. So basically what we wanted to see was whether there was competition or for this particular disaccharide. And this is exactly what we see here at 200 micromolar. Disaccharide is gentiobias is a heavy competitor for the flipping reaction. We have other disaccharides here. There is no competition at all. Now, this was telling us that the hydrophilic pocket may actually show high specificity for that particular head. We wanted to further show this, and what we did was to reconstitute in protoliposomes a glycolipid which has, instead of the 2 glucoses as in the head group, 2 galactoses. Now, these 2 sugars are very similar except for the configuration of one hydroxyl group. But what we see here is that even at 40 excess of these digalactose glycolipid, there is barely any competition. And even if we go to 100-fold excess, there is also a little bit of an impact. So this rather confirms that indeed the specificity for the head group is really, really high. And the lipid tails, most likely actually the specificity is not high at all. So okay, so all this was indicating, okay, most likely we have a trap and flip mechanism. I think it's pretty clear by now that it's very likely that the lipid will be loaded fully inside the cavity and that the credit card may be discarded. However, we wanted to do still something else. If you have a trap and flip mechanism, you need to go from inward facing to outward facing state. That's unavoidable. If you have a credit card mechanism, there is still the possibility that just one state actually allows the flipping of the lipid from one leaflet to the other. This is what has been shown for other flipases that use the credit card model. So to test this, what we did was to cross-link all the extracellular gates or all the intracellular gates, and then measure the activity of mutants which have been cross-linked. These are the solid lines that you see here, or non-cross-linked, which are the pointed lines, the dotted lines that you see here. So we see a proton transport. This is actually proton transport in this case. We see for the non-cross-linked mutants, we see activity, but when you have cross-linking of the gates, the activity is severely reduced. Now, this was definitely pointing towards a trap and flip mechanism, and I think all this information is actually summarized here, where we have binding of the glycolipid in the central cavity. I'm sorry, but actually there is a glitch here, and this graph is not showing actually as I had it in my Mac. But okay, the lipid is going into the central cavity here. And when it leaves the transporter, it will leave from either of the 2 gates. This transporter has 2 lateral gates. This was shown by also the genesis analysis that we did and the flipping assays. Either of the 2 gates is okay for the lipid to leave. Then you have binding of the protons to certain charged residues that we have also shown which one they are. And release of the protons, we believe, occurs through this transmembrane, through the gate lined by transmembrane helix 11 and 2. And in entry of the lipids, we believe it occurs through this gate, which is lined by transmembrane helix 8 and 5. So all this is also supported by our ND simulations of the inward facing state and the outward facing state. And we believe this could be actually a mechanism that may explain how other MFS lipases like MFSD2A work. And just this, I want to show you a couple of slides on the adaptation, how this protein actually helped this pathogen to adapt. Where I told you that in the hydrophilic pocket, we have this bunch of hydrophilic residues and some charged residues, right? You mutate those and we see an effect. But the very cool experiment that I want to show you is this one, right? We have here in the red curve, we don't have a proton gradient. This is in liposome. And you see this level of flipping. Now, if you have an inward directed proton gradient, we see flipping of lipids. The higher the proton gradient, the more flipping of lipids you will see. If you have an inverted proton gradient, then the situation reverses. So all this together with the metagenesis was telling us that this was indeed a proton coupled transporter, but how is this relevant for the bacteria? Well, the bacteria actually colonizes the human nasopharynx and the mucous membranes and the skin, where actually pH is around 5 and 6.5. So what we did was to make a knockout of this protein, and this is lane number 2 right here in the actual pathogen. And we go down in pH here in the solid media, and you can see clearly that the knockout of the protein doesn't survive. It doesn't grow at this acidic pH, just because of the activity of this protein. What this means is that when this bacteria is let's say in a clinical instrument where pH is neutral, then this protein is working slowly. There is still some proton gradient. There are still protons around. It's working slowly. When the bacteria colonizes let's say the human nasopharynx where pH is more acidic, this protein will work faster. What it means is that there will be more glycolipid on the extracellular side of the membrane. The polymerase will work faster, and more lipotequic acids will be available out there. This was shown here. We show it in the lab. We see more abundant lipotequic acids at a low pH than a high pH. We believe is that this bacteria requires more of this polymer when encountering acidic conditions, because these polymers are actually very rich in phosphates. And this may help to basically buffer the cell wall. Now, we don't know why would it need to buffer the cell wall. It may be because there are multiple different proteins important for this bacteria actually sitting at the cell wall. So maybe these proteins need to actually work at certain pH. And this is actually the role of adaptation of this protein. And this is actually all I wanted to tell you. And I want to thank again Bing and Elizabeth for the amazing work they did on this protein, and our collaborators, Jan Willem at the University of Lausanne, which helped us with making the knockouts in the pathogens, and Gerhard Huber and Reza at the Max Planck Institute of Biophysics helped us with the molecular dynamic simulations. And the funding for my lab comes from the Swiss National Science Foundation, the Helmut Koch-Stern Stiftung, and of course, I like to thank my institution, Biozentrum, in Università. Thanks for your attention.
We already see a hand up.
That was,
So, Professor Malata.
you were bad to me earlier. Thank you for this stimulating presentation. I was wondering concerning how do you measure this flippase activity? That's
yeah, this is what I left out, but
yes,
it's because of time. Yes, so I can explain to you, because actually it was a lot of work done by Inga and Elizabeth. So the substrate is not commercially available, the glycolipid. So we had to synthesize it. And not only that, but we needed a way to report whether it was inside or outside of liposomes, right? So what we did was to couple a fluorophore to the lipid tail of the substrate, and we synthesize it by purifying the protein that does the synthesis in the pathogen, purifying and then separating all the products by TLC, by the thin layer chromatography, and later on reconstituted this in a liposome. And the assay works like this. So if you have a liposome full with this now fluorescently labeled lipid, and you expose this liposome to a reducing agent, then only the lipids looking towards the outside of the liposome will be quenched, because the reducing agent will react with the fluorophore. If you have the flippase and you do the same, then because there is exchange of lipids, then more than 50% of the lipids will be quenched. And if you give enough time, this will quench even more. So this, of course, we analyze the product of the reaction by mass spec and also NMR. And basically the assay look like this. If you have empty liposomes, you will quench 50% of the population of lipids. And if you have the protein, the lipase, then more than 50% will be quenched. And this is how
that's made. That's very interesting. And one last detail: how do you check the sidedness of the protein? That's an important thing.
Yes, we did it because we had a construct coupled to GFP, and in between you have a TF site. And we, in liposomes, after reconstitution in liposomes, and we do this with extrusion. So we were pretty fairly, confident that most likely we have 50-50 population. We cleave the GFP outside, and so in this way it was confirmed. Yeah.
Thank you.
I was wondering with the similar system with the protoliposome, you can also measure proton transport.
Yes.
Because have you done it?
Yes, exactly. This is what I did. This is again another detail that is difficult to put in 20 minutes. Yes. So this assay, the proton transport assay, is this one right here. So we have protoliposomes, and now we have ACMA. And by establishing a proton gradient by addition of valinomycin, having a high potassium inside, low potassium outside, then we trigger proton transport. And what you see here is basically quenching of ACMA. And here you see the wild type. This is in blue, and you have here in black. This is empty liposome, basically. So yes, we use this proton transport.
Yeah, you mentioned, but was probably too fast.
Yeah, it's just a
but my question is, did I understand correctly that at the end, the conformational change which is imposed by the protons is independent of the presence of the substrate? It goes independently?
It's an antiporter. So protons will go from the outside to the inside in the absence of the lipid in the central cavity, because the lipid has been already released. But we believe that the presence of protons is important to go from outward to inward facing state. This will just facilitate this transition. But it works in an antiporter mode. Yeah.
I have more of a comment, a philosophical comment. I mean, I think it's fascinating. But I think the analogy with MFSD2A is actually the opposite, because in one case, you need the specificity is the single A salt. It's a lysolipid, and the A salt chain sits in the protein.
Yes.
Here, the specificity is the head group.
Yes.
It's the double sugar, which makes it such that it can't flip any bloody glycerophospholipid. So the A salt chain stays in the membrane.
Yes,
but it's the same fold, the same MFS, and it's completely opposite ways of dealing with the specificity problem. Am I correct in the way I'm seeing it? Or?
I believe, I mean, the amphipathic central cavity is there. It's
there for both.
That's exactly. But the specificity in the case of MFSD2A towards the lipid cell, may be absolutely different. For this particular, for LTAA, I don't think we have a specificity for the lipid cell at all.
Not at all. But and the lipid cell, the diacyl chain.
It's a diacyl chain, exactly. You have this is very different for MFSD2A.
And in 2A, you actually have a cysteine cross-linker, which to me is there with other charges to prevent that the diacyl goes.
Yes. The other thing is that in MFSD2A is in that case, it's a symporter, huh? This is a sodium-coupled symporter. So I believe there are, they overlap in multiple aspects, but there is still a lot of things to say about MFSD.
Another question is tangential. I mean, it's not the glycosyl transferase YDFP. So again, it's taking an activated sugar. It's taking it twice. It's taking it on the diacylglycerol. Does it have any resemblance to the ARNC GTRB? Like, is how you show it next to the membrane? How is it just peripheral? Does it have a transmembrane? What happens there? Because the typical reaction is with a polyprenol carrier and a phosphate. That's also,
that's
it's
also very different,
but the same. And I know it's not your main topic, but I help spotting.
No, this protein is actually very interesting, and also the whole pathway, because exactly, I mean, you don't use non-decarbonate pyrophosphate. This is a very different. Thiacylglycerol is very abundant in the membrane of this bacteria, so we believe that, of course, that the YPFP should come here to the membrane, and by a processive mechanism to do the synthesis. Now we don't know if this happens next to the lipase or maybe it's all around. This is not known. But yeah, this is
it's to be studied.
It's to be studied whether there is any interaction or what is happening.
Any other question from the audience here? No, let me check if there's something from the chat.
So given that we are about 1 hour late, I'm going to cancel the coffee break.
Oh.
But I mean, let's make it only 15 minutes long. And if you have questions for the speakers, you can, I mean, grab them over coffee. And I'm telling the public online that we're gonna have a 15-minute intermission from the first and the second session.
And we thank again the speakers of this morning. Qua che faccio? Dico, l'ho interrotta. Perché adesso riprendi con
Perché Now then, in order to catch up with the delay we had so far, we are starting now the second session, which is dedicated to the so-called eukaryotic world. So distinguished researchers in the field will tell us about their result on the structural biology of proteins that function in eukaryotes, including ourselves, of course. The first speaker of this session is Dr. Francesca Vallese, who's actually associate research scientist at the Columbia University Department of Physiology and Cell Biology. And she's got a very prestigious fellowship from ISNAF that selects the most promising young scientists to travel abroad and carry out cutting edge research. And before that, she was at Padua in our old friend Giuseppe Zanotti's group. And I'm gonna take no time more. And the floor is yours. Let's share the screen. Oh, that's it, I guess. And that's it. And that's it. Thank you.
Thank you very much, Beatrice. And thank you very much to the organizer for having me here. It's a great opportunity, and I'm very excited today because for the first time I will show our recent result on the structural characterization of the ankyrin-1 complex from the human erythrocytes. Sorry. Oh, thank you very much. So the red blood cells, also called erythrocytes, are the most abundant cell in the blood, and they are involved in the transport of the respiratory gases as CO2 and oxygen, of course, from the lungs to the tissues and vice versa. And they have this characteristic biconcave shape that has a surface to area ratio that optimizes this transfer of the gases, and are also very flexible because they need to pass through very small capillaries. The shape and flexibility of the red blood cells membranes is provided by a network of spectrin and ankyrin actin cytoskeleton. So here in this very beautiful negative stain image from Derrick et al. From 1987 paper, you can see this particular network in which you can see the spectrin and actin that are connected to the membrane by two different complexes. The junctional complex, that is the one that is responsible for the tetramerization of spectrin here, and the ankyrin-1 complex that is connecting directly the cytoskeleton to the membrane, and at the same time also mediate the clustering of functional related integral membrane proteins. And this last one is also the subject of my experiment, of our study, and also of the data that I will show you today. You need to imagine that what I'm showing you today from the erythrocytes is not present only in the erythrocytes, because we can find something similar in which we have a spectrin actin, a cytoskeleton, and the ankyrin involved in interaction, also in neurons, in cardiomyocyte, in epithelial cell, and skeletal muscle. So whatever we are showing here is something that you can imagine is present also in other cell types. The ankyrin-1 complex is characterized biochemically since the 1950s, so it's more than 50 years that we know, for example, some of the components of this complex. For example, Bentry, that is a transporter, that is an anion exchange of chloride and bicarbonate, that is always expressed as a dimer and seems to be present as a tetramer in the ankyrin-1 complex. Protein 4.2 that regulates the association of Bentry with the ankyrin, and the ankyrin itself, that is a 200 kilodalton scaffold protein that mediates association between the spectrin and the the membrane. And then glycophorin A, that is a single single transmembrane. A mutation in the protein involved is in this complex, in particular BEN3, ankyrin, or protein 4.2, are related with a severe kind of anemia called hereditary spherocytosis, in which the erythrocytes have this abnormal shape, are like a sphere and not anymore as a disc, and for that reason is called spherocytosis. So the spherocytes in this case are very fragile and are rapidly degraded by the split. If we can understand why ankyrin has this rule to connect the cytoskeleton to the membrane, why all these membrane proteins are keep all together, are like forming this cluster, one of the hypotheses that is also what we think is that ankyrin 1 complex form a sort of CO2 metabolome involved in the gas exchange. Because, for example, is known that carbonic anhydrase that converted the CO2 and water in a bicarbonate and proton bind BEN3. We know that BEN3 mediates the anion exchange of chloride, anion exchange of chloride and bicarbonate. That RH-Ag, that is another component of the complex, facilitate the diffusion, facilitate the diffusion and modulation on NH3, and is probably involved also in the diffusion of CO2. But even if we know so many things about this complex, we don't know exactly which are the components. If these components are all related to this complex, if are multiple complexes that interact together. We don't know the stoichiometry of the complex, how many of these components are involved. We don't know how ankyrin, that is a soluble protein, is interacting with the membrane, and how can manage the simultaneous interaction with the multiple membrane proteins, or even how this complex can be involved in the membrane curvature. To try to answer to all these questions, we decided to purify the ankyrin 1 complex, and since we don't know exactly which is the stoichiometry or the components, we decided to purify directly from the red blood cells. I will skip all the part that was pretty long for the optimization of the ghost membrane purification, or how we optimize and we selected the different detergents. But I will show you just the final purification protocol, because that, as you can imagine, was pretty tricky, because we have no any tag to purify the complex. So the first thing that we did was to solubilize the ghost membrane. Ghost membrane is the name of the plasma membrane once that you take out of the hemoglobin. Were solubilized with the digitonin. Then I did a step gradient with the glycerol to try to separate whatever was with a small molecular weight to something with high molecular weight. So, and I selected the bottom part of the gradient, in which I supposed to have the high molecular weight species. Then I did a gel filtration using a super 6 column, and from this gel filtration I obtained 2 different peaks, actually one huge peak, and what in my mind was a peak was more like a shoulder, but looking with mass photometer that allow you to determine the mass of this protein in your sample, we analyzed the first peak, the huge one, and the size was around 400 kilodalton. And we imagined that was BEN3 plus glycophorin A. And the second shoulder that we analyze always with mass photometer show us the presence of a huge complex at 1.2 megadalton. So we took this fraction and we froze a couple of grids, and we went through all the process for the collection of and for single particle cryo-EM. We collect more than 40,000 images, and here you can see one of the representative micrographs. This micrograph is covered by our particles, but I just selected a couple just to show you how it looks in terms of different views. These are the two disk classes from the analysis of these particles, and as you can maybe see immediately, is very dishomogeneous. So we have something very big in terms of size that I selected here in green, and something that is smaller than that, actually very small compared to the big complex that is here in red. The workflow for the processing is very, very complicated. I'm showing just a part of that. In particular, the first part in which from 800,000 particles, we have a first consensus refinement of what we call the core of the complex at 2.6 Armstrong. Subsequent analysis of this consensus refinement allow us to identify other 6 subclasses that we call class 1 to 6. There are different classes, but today I will talk just about class 1 and class 2, in particular because we think that the entire complex is the one presenting class 1 that is very similar to what we can see in class 3 and 6, that we imagine that are just complexes in which the transmembrane part was degraded or was just take out during the solubilization. Then we have a class 2 in which we have the presence of a different protein compared to the other classes that appear also in class 5. But just now I will show you first class 1. This nice movie was made by Kuk-Ju Kim, a PhD student in Oliver Clark Lab, and is a movie in which there is the map fitted with the model of the class 1. Here you can see the part that is the transmembrane part and the cytosolic part here in the outside. There are 3 band 3 protein, one in yellow, one in purple, and one in orange color. Then we have ankyrin here in red, and protein 4.2 here in green. In the upper part, in the transmembrane part that now we will see better from the top view, we have also the RH complex that we call the core of the complex. And you can see also the other 3 band 3 from the top view and glycophorin A. 1 glycophorin A each band 3 protomer. As expected, ankyrin has a central role in this complex that you can see here. In this image is missing all the transmembrane part except the RH, because I want to focus my attention just in the protein that are interacting with the ankyrin. Ankyrin has this particular folding that is called ankyrin repeat, that is formed by 33 amino acids with the 2 helices connected by a loop. For example, we can see that the ankyrin repeat region 1 to 5 here on the top is the part that is interacting with the membrane through an interaction with the RHCE, the N-C-terminal region of RHCE. This is the cytosolic domain of band 3, the one that we call band 3-1, and is not interacting with the ankyrin, but is interacting through protein 4.2 that is oriented vertically compared to the membrane and is interacting with the ankyrin region 1 and the ankyrin region 6 to 30. Then we have the second cytosolic domain of band 3 that we call band 3-2 that interacts with the ankyrin repeat region 17 to 19. And the third cytosolic domain of band 3 that is interacting with the ankyrin region 21 to 20. But let me talk a little bit about ankyrin because it's a very interesting protein. There are 3 ankyrin present in vertebrates, ankyrin 1, 2, and 3. Ankyrin 1 is the one present in the erythrocyte, but we can find ankyrin 1 also in the brain and in skeletal muscle. Ankyrin 2 and ankyrin 3 are present, for example, in cardiomyocyte and in neurons. Ankyrins share a common domain arrangement that consists in the 24 ankyrin repeat region that is also the part that is interacting with the other membrane protein. Then there is a spectrin binding module at that domain, and finally an autoinhibitory motif in the C-terminal tail. If we compare the structure that was solved before of ankyrin 2 here in gray with the structure that we solve in the complex of ankyrin 1 here in blue and red, it's clear that there is a dramatic change of conformation that involve the ankyrin repeat region 1 to 5. That if in the ankyrin 2 was the structure was completely straight, in this case the first 5 ankyrin repeat domain are going to be horizontal, perpendicular compared to the rest of the structure. And this new conformation is also the conformation that allow ankyrin to interact directly with the membrane, because you can imagine that in this part that now is available to the interaction, it's pretty easy for this part to interact with the membrane. If we compare the 24 ankyrin repeat region of ankyrin 1 from the sequence point of view, and we are going to we look in particular at ankyrin repeat 5 and 6, it's clear that there are some differences compared to the other ankyrin repeat sequence. In particular, in ankyrin repeat 5, there are 4 amino acids that are missing, and in ankyrin repeat 6, there are a couple of mutations that are not present in the other ankyrin repeat. It's very interesting to look at the conservation of the ankyrin repeat region 5 and 6 between all the 3 ankyrins, that also allow us to hypothesize that the same behavior that we are seeing for ankyrin 1 is happening also for ankyrin 2 and ankyrin 3. I don't know, here. So, and it's also interesting that looking more in detail, this is the structure of ankyrin 2 that I was mentioning before, in the presence of the autoinhibitory motif. To know. If we compare with the structure of ankyrin 1 in the presence of, in our case, of a peptide that is coming from the N-terminal of BEN3-1, it's important to mention that the peptide is oriented in the opposite, in the reverse direction to the previously observed ankyrin 1 motif. But looking at that more in details in this movie, you can see that here we have the cytosolic domain on BEN3. We have the N-terminal part that is going all along the ankyrin binding groove until arriving in this part that corresponded to ankyrin region 6 to 5 and linking also ankyrin 5 to 6. So what our hypothesis is that in the presence of BEN3 N-terminal domain, this replace the autoinhibitory motif and is going to bind ankyrin region 5 to 6. This allow the ankyrin repeat region 1 to 5 to change the conformation. And at this point, the change of conformation can allow the binding to the membrane. If we compare also here the 1 to 5 of ankyrin 1 and ankyrin 2 bind to the 2 different peptides, in this case RHCE, and in this case of NAV 1 to 5, well, 1.2, we can see that in this case they are binding more or less using to the same amino acid and in the same direction. Solving the structure of this complex, we were able to solve for the first time also the structure of the RH complex. And there are 3 RH protein in the blood, RH-AG, CE, and D. And in the RH complex that we solve, we were able to see that is an heterotrimer formed by 2 copies of RH-AG, here in blue and in light blue, and 1 copy of RH-CE, that has a very important role here because it's the one that is that connects the ankyrin to the membrane. The 3 BEN3 present in the complex are very interesting. Here I just aligned the 3 transmembrane part just to show you how is the difference in the position of the cytosolic domain. And it's also nice and very important to mention that in the case of BEN3-1, in this case, the orange one, that is the one bound to protein 4.2, the cytosolic domain is completely inverted compared to the one of BEN3-2 and BEN3-3. We solve for the first time also the structure of protein 4.2 that in this case plays a role as an adapter, stabilizing also the ankyrin interaction with the membrane, because as you can see here, protein 4.2 interacts with the BEN3, both cytosolic and also the transmembrane. Now I will tell you briefly something also about class 2. Class 2 was a little bit of a surprise because we identify an unexpected components of the complex that is aquaporine. So in this movie, in which you can see both the map and the model, you can see that is present the core of the complex, the RH domain, one of the BEN3, BEN3-1. There is protein 4.2 and ankyrin. And there is also this density that after we were able to solve at 2.8 Armstrong. And you can see that the fitting with the aquaporine is pretty. So I want to mention again that is an unknown participant in the erythrocyte ankyrin complex, and that is located in the interface of BEN 3.1, protein 4.2, and ankyrin interaction. And actually that is interacting with all these proteins. If you remember, I mentioned about the CO2 metabolome, and also aquaporine, since conducts both CO2 and water, makes sense if we think that is part of this CO2 metabolome. I mentioned at the beginning that the dataset was extremely heterogeneous, and we identify also something very small. Well, these small, very small components was a complex of just BEN3 and glycophorin A. In this case, I will show you just the transmembrane part. Should be present also the cytosolic part, but the resolution of this part was not very good. In particular, if you think how is flexible this cytosolic domain, it's not hard to believe that the resolution is not so good. So BEN3 is a dimer, here in light blue and in green, and is interacting with the glycophorin A for each protomer. And the glycophorin A, as I mentioned before, is a single transmembrane membrane protein, and is already known that regulates BEN3. So it was something that we expected to see. And is classically a dimer, but in this case is a monomer. And actually, if we try to put that as a dimer, but the second glycophorin can't be placed there because it will be completely wrong inside the membrane. Another interesting thing that we identify here in orange, that is in the interface between the two protomers, is a lipid PIP2. So I don't know if you know that, but PIP2 is less than 1% of the total lipid that you can find in the plasma membrane. And usually when you find PIP2, it is related with something functional, or for the function, also for the oligomerization of the protein. Here, this movie is just to show you that we place PIP2 there, but it's fitting perfectly, so there are no many doubts about it. But as this morning the professor mentioned to us, how we maybe can be sure 100% that what we see is something real, Well, we ask the same question to ourselves. So it's likely that in vivo, the three-band-3 dimers identified here, as well as aquaporin, are part of the same complex. And this complex is the total complex, or there are other parts that we can see just because we are solubilizing using the digitonin as a detergent. Well, to try to answer to this question in collaboration we prepared something that we call native red blood cell membrane. So I just sonicate the membrane. Is a very simple process. I sonicate the membrane, I extruded the membrane, and from the ghost membrane, I obtain these small vesicles that are native membrane vesicles. And in collaboration with Alex Nobel and Jake at the CMC, we started with some cryo-EM on this. Here you can see one of the tomograms that we collected, in which you can see these vesicles. And all around this vesicle, you can see a lot of protein, both in the inside part and the outside. We manually selected some of these proteins, and if you can see some shape of this protein that we selected, remember as something that we already saw in the cubic classes. But recently, we also were able to have a subtomogram averaging that reveals that the native complex present in the native membrane is very, very similar to class 1. Here is the subtomogram averaging in which we fit our model. And as you can see from the cytosolic part in particular, this fits perfectly. And also here, looking at the cut through, the cut through of this subtomogram averaging show us the presence of the RH domain, aquaporin, and the three-band-3. I know that I show you a lot of protein, a lot of data, but the take-home message is that we solved the structure of the erythrocytes ankyrin-1 complex, purified directly from the human erythrocyte, and that we identify also that aquaporin is a component of this complex. We identified that RHCE has a very important role because it's the one that mediates the interaction of ankyrin with the membrane, and that the RH complex is a heterotrimer. Subtomogram averaging of the particle that we extract from the native red blood cell membrane show that the endogenous complex adopts a similar architecture to that determined by single particle cryo-EM. So somebody today told me that is a happy ending story. So, and actually the story started in 2050. This is the first time that I tried to purify the ghost membrane from red blood cells. And this is 2000, and I was in Padua. And this is New York in 2021, the day of the data collection that gave us this nice data set. There are a few people that I need to thank. The first one is my PI, Oliver Clark. I'm sure that without Ollie, I will be still crying in front of this data set because it was a real mess. And the people in my lab, Filippo Mancia and all the lab of Filippo, they are really great person. And the people on the cryo-EM center that helped me in the data collection, Alex and Jake from the CMC, that we are having a great collaboration for the tomography, and Tito Cali for the University of Padua that I'm still collaborating because apparently it's very easy to have blood in Italy. So, and thank you for the attention.
Truly amazing, and many compliments to these exciting results.
Thank you.
And the session is now open for only 2 short questions. Shoot.
First thing, I don't think that is intrinsically unfolded. I think that is just very flexible because there is this connection between the transmembrane and the cytosolic part that is like 30 amino acids. And these amino acids are extremely, because you need to imagine that it is interacting with the ankyrin. I didn't mention that, but each domain that is interacting with ankyrin is interacting with a different part. So the interacting domain of BEN3/2 that is interacting with the ankyrin repeat 17 to 19 is completely different from the one that is interacting with the ankyrin 21 to 24 or that. So this is very particular, and I don't know if
that can be used for identifying or predict proteins.
So I think that at the level of so there are other proteins that are known to interact. Okay, in particular the ones that are interacting with the membrane are known. There is some ionic channel. There is some ATP, some other ATPase. So there is some other protein that is known. For what is I don't know if actually we check for the motive when we were looking, for example, at the neurons, at the ankyrin present in the neurons, but we were not able to identify this motive.
We have a similar question from the online audience. Dr. Saponaro from University of Milan congratulates as well and asks you, you showed that the 3 band proteins have different conformation. Do you have an idea of the physiological role of these different conformations?
Oh, well, it's very easy just to interact. So they are having this conformation just to allow the interaction of them. Yes. Yes, so I'm also very lucky because we collaborated with Filippo, and actually Filippo solved the structure of one of the malaria proteins. So yes, this is actually next step. Thank you.
Okay, I'm really sorry. I'm pretty sure that at the end of the session, people will flood you with questions. And our next speaker now is Professor Francesca Magnani.
Not yet professor.
Soon to be Professor Francesca Magnani from Pavia University. Actually, she did visit a number of different institutions after a degree in Bologna University. She was awarded her PhD at Trinity College in Dublin, and then worked at the LMB MRC with Henderson and Tate, and now is in Pavia working on flavoenzymes, which are Pavia proteins, I would say, but also heme membrane proteins. And today she is gonna tell us about work carrying on.
Yes, some old story and also sorry.
Now then, interrompi on this. Let's just open your talk. What the hell? Escape. Close this, and we get to which one is yours? Brilliant. Come on. Okay, now we go back here and we condividi schermo. And we here, condividi, and we do this. Okay, and we also do this. The floor is yours.
Thank you very much. Thank you to the organizers for the invitation. And so today I'm going to give an overview of past work on protein coupled receptors then going to a more recent work on NADPH oxidases, and then ongoing work on absolute carrier. So, okay. So G-protein coupled receptors, actually yesterday we heard both Bernadette and Alexis mentioning the 8-way receptors and mutagenesis studies carried on this. So it's just timely perhaps to remind of the kind of work that was carried out in the laboratory of Chris Tate in Cambridge at the LND-MRC. And also a bit of an historical point, this work started in 2005, and at the time there were, we saw a few times the histograms reporting the number of membrane protein structures solved, and this, the overview was actually even sadder for G-protein coupled receptors, as during that time only rhodopsin had been solved. This is the largest family of proteins in the human genome, and are very important for several biological processes. So they have always been considered very highly sought targets for drug discovery. Unfortunately, the paucity of structures hinder that progress. So around the time, many groups started to think creatively on ways to solve the problems. The problems encountered with GPCRs are actually encountered for most membrane proteins, which is well, poor expression, I didn't put it here, but poor stability in detergent solution, and conformational heterogeneity, which in the end result in difficulties to purify and crystallize. Again, this was pre-cryo-EM time. Crystallize the target. These receptors are very intriguing. They have a beautiful pharmacology. They are able to bind also many different types of ligands from a chemical standpoint. So going from photons in the case of rhodopsin to neurotransmitters to small proteins. And the binding of the ligand on the extracellular milieu elicits a conformational change on the intracellular side of the protein, which opens up the binding site for intracellular effectors such as trimeric G proteins or arrestins, and then they exert the specific signaling in the cell. So the way that Chris decided to tackle this problem actually got inspired from a previous work in the lab of James Bowie, where he demonstrated that it was possible to mutagenize residues of transmembrane protein in transmembrane helices without perturbing dramatically the function and making them more stable. So we wanted to achieve this. We wanted to demonstrate that this was possible to do on a larger scale. So we embarked on a massive mutagenesis of 3 different GPCRs, and the scope was that to evolve at the same time the stability, so to find mutations that would increase the stability in solution, but also give a more stable receptor in one specific conformational state. This was achieved by actually the method, once it was tested a few times, is quite simple, and this is also part of the beauty of it, and very robust. So we performed a comprehensive alanine scanning, so from residue 1 to residue 300 and so. Then the receptors, the mutants were transfected, and the crude lysate was subjected to heating, incubated before with either an agonist or an antagonist. And this was very important because, again, we wanted to obtain mutations that stabilize one specific conformational state. And then we obtained the melting curves that we are quite familiar with. So usually one mutation is not enough. One mutation is not enough to achieve enough stabilization for crystallize the receptor or purify it efficiently. So this was an iterative process. Our golden standard again was the TM of rhodopsin, which was about, with this type of assay, resulted to be about 55 degrees. So anything that brought us above that was considered acceptable and brought forward to crystallization. So this is just some results to exemplify this work. And this was our data from the adenosine 2A receptor. And again, it was not surprising, but pleasing to see that indeed different mutations stabilized the mutations that stabilized the active state were distinct from the ones that stabilized the inactive state, the antagonist bound state, with the exception of three, in this case, of three positions, 3 amino acids that when mutated stabilized to alanine, stabilized both conformation, but this was an exception. So after some more work, two structures of both the active and inactive site, receptors were obtained, which allowed to start analyzing the binding sites, the binding pockets, and changing occurring when an agonist is bound compared to when an antagonist is bound. And there is the agonist tends to bind in a more profound, more in-depth in the binding pocket compared to the antagonist, which is slightly more shallow. But the biggest change of all, which is observed, is the movement of the of the bottom part of the transmembrane helices, with particular swing out movement and opening up of the bottom part of the helix 6. Without going into further details, obviously this type of work had profound implications also to enable drug discovery. And for this purpose, it was created already in 2007 a biotech branch from our work, Heptares Therapeutics, which brought forward this work, and now their pipeline looks populated by many ligands in different stages of preclinical and clinical studies, with 3 drugs that are on the market. Then we move forward to another type of protein, which is different from the ones that we talked about during these 2 days. These are membrane enzymes called NADPH oxidases or NOXes, which can be considered professional producer of radical oxygen species. It is a family of 7 members. They differ for the number of proteins that associate to heat and contribute to the regulation of this enzyme, which are tightly regulated, with the exception of NOX5, which is a standalone enzyme, and for this reason is the one that we decided to focus on. Even though they differ in their regulation mechanism, these enzymes all share a common catalytic core, which is formed by 6 transmembrane helices transmembrane domain, followed to the C-terminus by a dehydrogenase domain, which is in the cytosol. The dehydrogenase domain hosts the binding site for NADPH, which is the initial substrate for catalysis, which donates an electron to FAD. And from here, the electron is then passed on to 2 chemoprostatic groups that are coordinated by 2 couples of histidines in the transmembrane helices. And finally, the electron from here can reach the other side of the membrane and is accepted by the final acceptor, which is oxygen, forming superoxide, with the exception of NOX4, which forms hydrogen peroxide. These enzymes are found in the human body, are found in many different tissues, but the one that has been studied the most already from a few decades now is NOX2, because is involved, mutations in this enzyme cause a genetic disease called chronic granulomatous disease. This NOX2 is present in neutrophils, macrophages, and monocytes. So mutations of NOX2 cause a failure of the innate immune system, and so patients suffer from infections of bacterial and fungal infections that normally would not cause particular problems, and sometimes with dear consequences, undergoing death. NOX5 is interesting also because it's quite well conserved across different kingdoms. So not only is present in vertebrates, but is the NOX present in plants. So plants contain, for instance, Arabidopsis has 10 copies, 10 different genes of the homologue of NOX5 called RBOH, which are located expressed in different tissues. And their role seem to be like adaptation to stressors such as high salinity in the soil. And they're also present in cyanobacteria. And sorry, I'm going to the prokaryotic world for a moment, because Simone Nenci, who was the postdoc working in Andrea Mattevi's lab at the time, started this project, and he found he tested three different genes in cyanobacteria, and at the end chose the one from Cylindrospermum staniale because it expressed better, and it has actually the highest identity to the human NOX5. However, it wasn't simple, so it started to work on the isolated dehydrogenase domain, because I have to say that this enzyme, when you start to purify them, they proteolyze massively. So at least in our hands, we had great problems of proteolysis. So the decision was, the divide and conquer approach. So let's learn from the individual domains as much as possible, and then hopefully one day learn how the whole enzyme works. So he started to work on the dehydrogenase domain, which is homologous, belongs actually to the family, to the largest superfamily of ferric reductases, but he could not purify. I mean, this domain was quite unstable. It did not retain FAD, which is considered bad news usually in purification. But then, while he was testing different constructs that he made to express this domain in coli, he found one that, when it started purified, was yellow, bright yellow, indicating that the FAD was retained. Then he managed to crystallize it, and then it became obvious why this particular construct gave higher stability and yielded crystals, yielded a structure. Because basically there is, as reported at the bottom of the figure, there is this short extension, PWLEL, downstream of the last native amino acid of NOX5, and particularly is this additional tryptophan that plays an important role in stabilizing the domain. And you can see that the tryptophan stacks beautifully against the FAD moiety, stabilizing it. Indeed, sorry, this is an unwanted effect, but in fact, the mutant had an increased melting temperature of about 19 degrees, which is quite massive. However, this variant, this mutant, was much less active compared to the wild type, about 6 times less active. And again, looking at the structure, we understand why this is the case, because basically, if you look at the image, the insert on the right-hand side, where these additional amino acids have been removed, and you see in gray, in dark gray, where the native C-terminal residue, the last phenylalanine, is placed, you can appreciate the fact that the additional amino acid actually causes steric hindrance, occupy the binding site for NADPH. So basically, it acts as an inhibitor, and by occupying the binding site for NADPH, the catalysis cannot begin. So the enzyme is much less active. Then we moved on to the other part of the catalytic core, which is the transmembrane portion. And we managed to obtain crystal by using lipidic cubic phases, which is another one of these new methods that came upon during these dire times in the early 2000s, which was really a game changer for small membrane proteins to obtain crystals. So we crystallized this domain, beautiful red crystals. The solution, the structure was solved to 2.1 angstrom, making use of the anomalous signal of the 2 irons within the heme. And then we could start building a crystallographic model of the catalytic core having the two domains, and to appreciate the beautiful geometry that is imposed by this structure to the substrate, the cofactor, and the two heme groups, which is ideal for shooting electrons from one side of the membrane to the opposite one, to the final acceptor, oxygen. Also, we think we identified the oxygen binding site in the structure. This site is occupied by water molecule, and this pocket is formed by hydrophilic residues, arginine, histidines, and this residue, and also the propionate, one of the propionate chains of the porphyrin ring. And these amino acids are actually totally conserved across NOXes. And mutations of this amino acid are actually among those that cause in NOX2 chronic granulomatous disease. We also made this mutation in NOX5 of these residues, and indeed found that the enzyme is much slower, doesn't work as well. The mutations of these residues basically do not allow the enzyme to undergo reoxidation. So it cannot function as efficiently as the wild type. We grafted the mutation that I described for the hydrogenase domain that allowed to crystallize that domain, the tryptophan mutant. We grafted it onto the full-length enzyme, and it was interesting to see that this stabilization was transmitted was no longer a local effect on the pH domain, but was transmitted also on the transmembrane domain. We established this by monitoring the retention of heme in our stability assay. Heme is one of the first things that goes when purifying the full-length enzyme. And the stability was lower than what observed for the hydrogenase domain. 8 degrees nonetheless was quite remarkable, and is also noticeable when looking at the SDS page gels, where in the wild type we have a band at the top caused by aggregated protein. This is no longer visible or is much reduced in the mutant version. So for quite some time we were stuck there because again we did not manage to solve the problem of proteolysis. So the amount of protein that we could obtain was very little and very dirty, non-homogeneous. But then we obtained an antibody from our colleagues in the University of Braunschweig, Federico Bertoglio, in the lab of Michael First. And this antibody has a nice property of allowing us to purify for the first time to see in gel a very nice band of the full-length NOX5 together with the antibody, which is at the top. However, the protein is still, the preparation is still quite polydisperse, and it carries NOX5 is stuck to the NAK, which usually is not good news because this is a heat shock protein that usually that binds to unstructured regions unfolded regions of proteins. However, we were very intrigued by this because the activity of these fractions is extremely high. So the NAK does not seem to cause problems in that sense. And for curiosity, we put it on a grid and did some preliminary cryo-EM studies. And unfortunately, it is possible to see perhaps from these models for these reconstructions that the sample is either very heterogeneous or there is great flexibility. Anyway, so there is still work to do, and is work currently in progress. And then in the last couple of minutes, I'm just telling you about the recent work started on a heme exporter called FLVCR1A, a very complex name, which is actually an acronym of feline leukemia virus type C receptor, because this transporter is the receptor for this feline gamma retrovirus. but it is known to be involved in 2 genetic diseases in humans, PCARP and HSAN. Both these diseases caused by mutation on this gene are the phenotype basically is on the damage is on the neuronal tissue, even though this transport, which is interesting because this transporter is ubiquitary. And when the knockouts mice reveal that this transporter is essential for erythroid maturation and cause early death of the embryos, probably due to hemorrhages, which are quite clearly seen in these images, due to the fact that when this transporter is no longer present or it doesn't function, heme is accumulated in the cells. Free heme is very toxic and cause cell lysis. Also, these mice showed skeletal abnormalities, which is also quite interesting. So there is very little known about this transporter. The mechanism of transport is not characterized, but first it was necessary to purify the protein. And the first thing that one usually does is to establish where to put the tag. And indeed, the tag at the N-terminus is not tolerated, so the transport is not working efficiently, and also that the tag at the N-terminus causes a accumulation. I didn't put the images here, but if you look at the cells under the fluorescent microscope, you see an accumulation of protein in the endoplasmic reticulum. So we set for the tag, the construct with the tag at the C-terminus. The protein is glycosylated, as one can see from the digestion with PNGase F on the left-hand side. And then we started performing a pre-purification screening to decide which detergent was best to use. And luckily, of the panel shown here, only sodium cholate was really evidently bad, but we had a few that could be used. So the first purification was actually not perfect because there is a lot of protein that is aggregated, as one can see from the void volume in the chromatogram, and also the protein at the top of the gel. So we went back to use this purified protein nonetheless to characterize better the detergent, buffer conditions, salt, and so on, and various additives. So we repeated the detergent screening, found some detergents that were clearly not good on the top left corner of the image, and but some that were promising. And then we tested the lipid additives, and with DDM worked very well to add the cholesterol hemisuccinate, whereas interestingly, addition of either cholesterol hemisuccinate or phospholipids in the form of azolectin with LMNG did not improve this particular preparation. And so with the finalized buffer conditions, this is the purified, the results for the purified protein, which is active because it's capable of binding to the substrate, coproporphyrin and heme. Coproporphyrin is a catabolite of heme. This experiment was done using microscale thermophoresis and also corroborated by nano-DSF for heme. So even though I do acknowledge that this was a very optimistic experiment to carry out, because this is a very small protein, it's a 65 kilodalton. So we made grids and put it under our glaciers. And the two classes are actually not bad, although there aren't, immediately appreciate that there aren't prominent features, and the 3D reconstruction even so shows some density inside the nanodisk. So at the moment we are screening antibodies and nanobodies to circumvent this problem and hopefully improve the structural work. And with this, I'm just going to show this slide to thank all the people I've worked with for the work that I talked about today. So people at the LMB, Chris Tate, Richard Anderson, Geber Scherpler, and also my bench workmates, Yoko Shibata, Maria Serrano Vega, and Anthony Ward. And then at the University of Pavia, the groups of crystallography, headed by Andrea Mattevi, Simone Nenci, particularly for the work on NOx5. And then for the work on the transporter, my acknowledgements go to Luigi Scetti and Adriana Giorgiana Pragica, a brilliant student. And of course, thanks to Andrea Alfieri, who is the manager of our electron microscope, and collaborators in Turin and also at the University of Braunschweig. And I leave you just with this slide just to show the pipeline that is at disposal. We're very happy about this, and I think it was the result of coordinated efforts from the different groups in the crystallography division of the department, so to say, and also some money infusion, which allowed the acquisition of an electron microscope at 700 megahertz NMR, super resolution microscope, and so on, which came from the Ministry of Education program of Department of Excellence. So hopefully these programs will be potentiated, and because I think they were really positive. And with this, I'm thanking you for your attention.
Thank you, Francesca, for the beautiful result you showed, and also for the description of the difficult yet extremely interesting path that one has to follow to strive towards results. So they were both informative on this side and also on the results you got previously. Thank you so much. Other questions from the audience? Any questions? Please.
Okay. Thank you very much for your presentation. I was wondering, when you tried additives, did you perform the whole purification again, starting with the additives, or did you do the purification and then you added the CHS azolectin? And the second question, did you try azolectin with DDM?
The first question, so no, to be faster, the little protein that could be purified with the old protocol was used then to test these additives. And azolectin with DDM, I'm not sure. I think we did. And the second point, I should look back. I don't think my recollection is that it did not improve, but I should look back onto that. Yes. Thank you.
Okay, thank you.
Okay, so if there are no further questions, I would call Anna Moroni. Now, just a brief presentation of Anna Moroni, who really doesn't need the presentation. She's working on ion channel biophysics in the University of Milan, and she really does everything, I would say. So she would do biophysical characterization, but I mean also in cell work and functional work, and of course, I mean, she couldn't miss on the structure of those amazing
No.
molecules. So thank you very much
Thank
for
you.
coming, and
Thank
please.
you. Thank you for this beautiful occasion, friendly environment, great talks. I'm so happy to be here. So I'm going to talk about the structure of an ion channel, which is called HCN4, which is also mentioned as the pacemaker channel in the heart. So a short introduction on the cardiac pacemaker. The automaticity of the heartbeat starts from the sinoatrial node, and the electrical impulse is then transported through to the ventricles for contraction. And the sinoatrial node myocytes, of which you can see one represented, they are autonomously beating, and because they are constantly firing action potential. And the important issue is that there is this diastolic depolarization phase that leads to the firing of the next one, which gives the automaticity. And in this case, the current which is producing the depolarization is called funny current, because it is activated at negative voltages, and it's unique among ion channels, and is also in short called IF. IF was identified and characterized by Dario Di Francesco in Oxford in the 70s. And since I was a postdoc of Dario in the 90s, I also get infected by this passion for the current. The current is also important because it's regulated by cyclic AMP, and therefore can control the rhythm of the heartbeat. Because if you increase or decrease the current, you change the slope of the depolarization, and therefore you accelerate or decelerate the heart rate. And this is related to the regulation of adrenaline acetylcholine on intracellular cyclic AMP, which then affects the current. The molecular determinants of the IF current are the ion channels called hyperpolarization activated cyclic nucleotide regulated, in short HCN. Shown here, they are tetrameric. Each monomer contains 6 transmembrane domain that bears the voltage sensor, and then they have the cytosolic cyclic nucleotide binding domain. They pass both potassium and sodium, and they have one known drug on the market, which is ivabradine, that is a pore blocker, and it's prescribed for angina when it is necessary to decrease the heart rate. And the only problem is that ivabradine is not specific for this subtype, the cardiac subtype, which is HCN4, but also affects with this very general mechanism of pore blocking the other subtypes of the HCN, which are expressed in the central and in the peripheral nervous system. So you can imagine the side effects. So as I was mentioning, the cardiac subtype is HCN4 and resembles very closely all the properties of the funny current. It has this slow time-dependent activation, the strong activation by cyclic AMP, which is better shown here in the activation curve of the channel that shows how negative voltages increases the open probability, and how the cyclic AMP addition shifts this voltage dependency to the right. We express this as delta V1/2, which for HCN4 is about 18 millivolts, meaning that, for instance, here at -75, you have a completely increased open probability that is then generating an increasing current. So obviously we, with the cryo-EM era, and with the luck that I was in Colombia in the lab of Harry Colcraft, that by chance was the neighbor of Filippo Mancia, we decided to purify the protein and to try to do the structure by cryo-EM. And the very beginning, I'm very proud because I was myself producing the protein, making the protein, which was was a sabbatical. So after many years, I went back to the bench, and it was a fantastic occasion. And of course, the rest of the story was carried out by Andrea Saponaro, which is a researcher in Milano, with whom I strictly collaborate for many years. So we solved 3 structures of ACN4, 2 in the APO configuration and 1 in the holo configuration, and with different detergents. This is LMNC CHS, so these are amphipols, a different resolution, as you see. So this is the protein. It's as I said, is a tetrameric protein formed by identical monomers with the transmembrane part and the soluble part shown there. This is the view of the pore from the top of the membrane, and this is the monomer that contains the transmembrane domains, the voltage sensor, the pore unit, the C-linker that connects the CMBD directly to the pore. And there is another ACN domain, which is shown here, which is soluble. And this is the way in which the domains are swapped in the cytosolic part. As you can see, the C-linker of the green subunit is below the transmembrane part of the adjacent subunit. So, sorry. Now, what we learned from comparing the holo and the APO structure is the pathway, are the conformational changes that in the cytosolic part are induced by cyclic AB binding to the CMBD. And as you can see, there are several movements, but the most important one is that one subunit affects the position of the helices A prime and B prime of the adjacent subunit, the green one. And it is like lifting up this, part of the C-linker of the adjacent subunit. And this is very relevant because in this position that is at the interface between the C-linker and the transmembrane portions, we found the presence of what we call a tetrad, an arrangement of 4 amino acids that are coordinating a ion that can be placed in the extra protein density. And these 4 amino acids are aspartate, 2-histidine, and a glutamate. And they are a metal ion coordination. They create a metal ion coordination site, basically. And this, it's actually, to my knowledge, the first time that in an ion channel, it's found such a similar structure that basically connects the transmembrane part to the cytosolic part of the protein. Being the cytosolic part regulatory, because it's the cyclic nucleotide binding domain, we immediately imagine that it could play a functional role in connecting the 2 functions of the channel, the voltage sensing part and the regulatory part in the cytosol. But first, we wanted to verify that this ion could be a relevant, physiological relevant ion like magnesium, which is abundant in the cytosol. And so we tested the possibility that this ion was a magnesium by thermal shift assay, which has been mentioned many times in these 2 days. And here we use GFP, the survival of GFP fluorescence, as a readout in gel filtration. And you can see the stability of the protein in normal condition in which we purify the protein is about 55 degrees. But if we remove magnesium with ATTA, we have a destabilization of the protein. And so we assume that they are losing this binding and this connection, the protein. And then if we add magnesium, we reobtain the stability of the control. The other step that we wanted to do is to verify if this magnesium could have a functional role in the protein. And the guess was that it could facilitate the action of cyclic MP in affecting the voltage dependency of the channel. And basically, we were checking what was the shift in the activation curve, the delta V1/2, if we were mutating the amino acids or if we were providing ADTA. And we found that indeed there is a decrease compared to the wild type channel if we introduce mutation in the 4 amino acid that generates the binding site. And this is one histidine, this is another of the 2 histidines, this is the aspartate. The glutamate doesn't go to the membrane, so we could not test that mutant. Double mutation, the triple mutation, they all have basically the response to cyclic AMP, and ADTA mimics the mutations. They have, but they are not completely removing the effect of cyclic AMP, because in order to remove totally the effect of cyclic AMP, we realized later that we had to disconnect another link of the cytosolic site to the transmembrane part with the 3rd unit, which is shown here in purple. And this is another bond that if we mutate one of the 2 partners together with one of the component of the tetrad, we completely abolished the effect of cyclic AMP on the gating of the channel, the voltage gating. So at the end of this investigation, we could support the hypothesis that the transmission of the cyclic AMP binding to the voltage sensitivity of the channel doesn't move via the C-linker, which is connected to the pore, as everybody was thinking, but in reality moves through a conformational changes that doesn't pass directly to the pore, but goes on this in this way to the voltage sensor through the interaction that we have been showing. And this confirms a large number of results, including previous results from our group. So the other feature that we have been analyzing is the transmembrane part of the protein. And as shown already previously, because McKinnon was obtaining the structure of HCN1 in 2017, also HCN4 has this parallel arrangement of the 3 transmembrane domains. There's 6 and there's 5 that belongs to the pore, and there's 4 which belongs to the voltage sensor. And they are connected by a large number of hydrophilic and hydrophobic contacts. So we had obtained structures of the holoprotein in which this parallel arrangement is even apparently further stabilized by a few lipids that we find, like for instance this lipid that surrounds S5, the lipid in yellows that are stabilizing the contact between S5 and S6 with the pore, and one lipid that seems to stabilize the S4, which is the voltage sensor. And they are also, and in this case, there is this link between asparagine and an aspartate, which we can see in the structure, glutamate, sorry, that we can see in the structure. But we also obtain a structure that has no lipids, and in this case, the bond is broken. There is a apparent turning of this S6, as shown by the position of this glutamine here, that is not obstructing the entry of the pore anymore. And so we had symptoms of the fact that the pore could be open in the absence of lipids. In particular, from a side view of a monomer, we could show that there is a partial or a complete detachment of all the zipper of hydrophobic and hydrophilic links that keeps bond S4 and S5 in the parallel arrangement that I was showing you before. So this detachment allows S5 and S6 to rotate, and this is presumably occurring like this, and is presumably opening the pore of the channel. Of course, to have an open pore of this channel is a big deal, because they are activated by voltage, in particular by hyperpolarizing voltage. So at 0 millivolt, which is the condition of cryo-EM, the pore would be always cannot be obtained in the open configuration. But with this lucky strike of the delipidation, we thought we had an open pore. And indeed, this is the profile of the diameter of the pore, which is open in 2 of the 3 structures and is closed in the 3rd one. And exactly this Q519 generates a strong constriction at the cytosolic site, together with other with these 3 hydrophobic amino acids that are turned and removed in the supposed open structure. So we did, of course, MD simulations, and we started to validate the fact that the cytosolic side was open and accessible to water and ions. And then we perform longer simulations in which we could see ions passing through the filter. The blue are the potassium ions, the yellow are the sodium ions. There are either 2 or 1 ions in the filter, and the entry of a second ion knocks off the first ion. This can be also generated by a sodium ion that is able to knock off a potassium ion. So with this molecular dynamic simulation, we could reconstruct very interesting features of the permeability of the channels. As I was mentioning, the selectivity filter can be occupied by 2 or by 1 ion, alternatively. But the interesting part is that when 1 ion occupancy occurs, the position of the ion at the plane of this carbonyl oxygen had never been observed before, because this is actually, usually the ions are located into cages of 4 carbonyls, but at the plane of 2 carbonyls was never been observed for potassium ions. It was observed for sodium ions. And this, it's actually very difficult, because the potassium ion has a larger radius than the sodium ion. So it's also strange that the same position, that the same binding site can host both. And in fact, in order to host a potassium ion, the carbonyl plane has to be cannot stay. It has to distort a little bit to allow the occupancy of a larger ion, while the sodium can enter without distortion. And this conformational change of the filter, we believe that can be related to the known low conductance of the channel, which is in the femtosiemens range, which makes it the smallest ever measured ion channel. This is an achievement of Dario Di Francesco, that was able to measure such a small current. And another characteristics that we explained with our molecular dynamic simulation is also the low selectivity for potassium of the channel, that has many of the features of potassium channels, but in reality can also pass sodium. And it has a known selectivity 4:2, potassium to sodium, which is, let's say, arithmetically explained by the fact that sodium by itself cannot permeate, but only can permeate in the presence of potassium. So given that this configuration of ions is never found, we can explain also the this kind of selectivity for potassium and sodium. The last thing that I want to touch is the story of ivabradine, which, as I said, is the only drug on the market that targets HCN channels. And when we had the open pore, we decided to do a docking of ivabradine. As you can see, it cannot be docked inside the pore in the closed conformation, but it can be docked inside the pore, even though it can acquire many positions, but one was, I'm showing one of the most, with the lowest energy that we found. And at this point, Andrea Saponaro decided to do the structure of the channel with the ivabradine inside in the presence of ivabradine, with the hope of finding it in the open pore. And this is now very new, recent data of Andrea that has been able to see a non-protein density in the pore. This is without symmetry, is in C1, and the resolution is not too bad. It can tentatively model the ivabradine molecule inside. And of course, there are three other small densities because the protein is tetrameric. So tetrameric without imposing the symmetry. And in fact, there is probably a preferential position, but there are the ivabradine can assume also three other positions with lower occupancy, of course. So on this basis, he decided to apply to impose a C4 symmetry in order to improve the density. And he had improved the density to the point that now it can fit ivabradine quite well. In particular, this linker, flexible linker between the two aromatic rings is well positioned. And the interaction site of ivabradine are asymmetric. This is the trimer, one of the four subunit has been removed, and as you can see, there is a ring, hydrophobic ring of tyrosines that are interacting with the ivabradine. There is another group of two hydrophobic isoleucine that interact with ivabradine, and there are also two cysteines on the top that belongs to the selectivity filter that are interacting with ivabradine. So altogether, these are all hydrophobic interaction of the molecule. And as you can see, they are asymmetric, which shows why there is a preferential pose of ivabradine in the densities already in C1. And with this, and these amino acids that have been identified as interacting with the drug, they were known to reduce ivabradine efficacy in the channel. These are mutations that were done by the lab of Dario Di Francesco a few years ago, who had also identified that the tyrosine and the isoleucine are reducing much the effect of ivabradine. This is, let's say, the wild type effect of ivabradine that basically reduces, brings to zero the current. This is the reduced effect of ivabradine on the two mutants, and the double mutants zeros the effect of ivabradine because the shifts the affinity to millimolar. So with this, I am coming to the conclusion. I hope I was fast enough for everybody to catch the trains. So the conclusion that we can draw by this structure of HM4 are that there is a connection between the transmembrane and the cytosolic domain, which is relevant for the transmission of the effect of cyclic MP to the transmembrane portion where the voltage sensor is. And then there is a mechanism of pore opening that has to do with the detachment of the voltage sensor S4 from the pore unit S5 and S6, and we have luckily somehow obtained this by protein delipidation. And with the open pore, we could describe many of the features known for the current or for the channel. In particular, the low conductance and the weak potassium selectivity. And then the latest, which is the position of the ivabradine in the binding site inside the pore, which we hope will open some approach to identify more subtype selective drugs for this class of channels. And with this, I just want to mention and to thank Andrea Saponaro, which is the person that has been basically done most of this project with the help of the cryo-EM facility of Milano and with the help of Alessandro Porro, which does the electrophysiology. And of course, a great contribution from Columbia University, because we started there the project and Hunter Gies was helping us at the beginning. And also to the group of Darmstadt for the molecular dynamic simulation. And thank you very much for the attention.
Thank you, Anna. That was beautiful. And the obvious question to me, do you already have some suggestions for more specific binders with respect to ivabradine?
The obvious answer is unfortunately not, because the pore is so conserved among the 4 subtypes that is basically not possible. But since we have other changes in the open pore, and we see a change in the extracellular part of the pore of HN4 compared to HN1, we want to tackle that.
To investigate
point,
that.
but not as pore blockers, as from extracellular side.
Sorry, one short question from the public. Call of the train.
Anna, those other densities next to the drug.
Yeah.
Are you sure you think it's other conformations? Are you sure it's not a lipid-mediated interaction?
Inside the pore?
Yeah. I just saw.
Yeah, well, it makes sense since the protein is tetrameric and the binding site is
one. It can be one of every 4.
It can be
in each protomer.
Yeah.
Yeah.
See, so you're agreeing with me.
So it's you're seeing the 4 possible conformations of one drug binding.
One preferential where the density, but there are also because the molecule can easily, because it's symmetric, the tetramer, so it can easily be positioned in any of the 4 possible binding sites.
Yes, but if it's a homotetramer, you should only see one, which is why you don't see the other densities if you do see 4, correct?
No, yes, you have 1 per pore, but when you average, you have 4 with different occupancy, different probability, but you have 4.
Beautiful.
If you have 1 binding site inside and you're averaging all the particles, it can be in any of the 4 possible corners, let's say.
No, I was just wondering if it could be that there's some lipid in there as well for whatever reason. I doubt it, but I'm just. No.
I have to ask Andrea. I actually don't know how they could exclude the possibility of the lipids, but because the densities, they fit very well ivabradine. They have this a little bit of a turn.
All of them.
Well, some more better.
Possibly they didn't see the density without ivabradine.
That's great.
Without ivabradine, do you see the density? No. Without, we don't see it.
No, don't see it. Thanks.
That's right. Yes, the lipids could occupy the without. Grazie, Beatrice. Yes. I should, but I don't know, because we have done the docking. For the docking, you mean? Yeah, exactly, because they always tell me, I don't trust this. Okay. Exactly. And molecular dynamic simulation, we can extract the contribution of the, now that we know the bind. Okay, thank you.
Now then, it has been a pleasure and an honor to have all of you. I would like to thank the speakers, all of them. Please. I would like to thank Filippo and Margherita, with whom we organized the workshop. I would like to thank all the Prometeus consortium for their support, and of course the audience who is in presence and who is unfortunately at a distance, but we had very good attendance, and I thank, of course, the speakers for providing such beautiful, interesting, and also the public for very interesting and stimulating discussion. Now, I would like to keep you here for longer. We should have had half an hour of discussion, but I understand that some of you are missing trains and flights. So I do suggest that those of you who want to continue to discuss and interact can do it in the foyer. We have spare coffee, juice, and cookies. But I would kindly ask the Prometeus gang to stay here for our project meeting. And thank you for everything. Okay, we have more.